Hey, everybody. Happy Wine Wednesday. Uh, I'm sorry, I've got my glass turned the wrong way. Got my little seahorse glass that you can barely see. Holly is with me. Ray is with us, but being silent. Chris Carey is actually driving to a better location so that we can hear him better because of his internet connection. And we have special guest Graham Short with us. Oh,
Hi
God, what's
there.
going on here? Hang on. Oh, I see. Okay, sorry guys. I had my YouTube playing in the background. I hear me in the delay, and I'm like, what the
Oops.
heck? Okay. Anyways, Holly, real quick, what you sipping on? I see Zen Ginger already.
Oh, so I went the NA route tonight. So I
Oh,
have a
okay.
Fanta float with huckleberry ice cream in
Yum, nice.
Hey,
it.
hey. All right. And Graham already was mad at me because he said he forgot his wine, and but we're good. Okay,
Also nice.
so guys, we've had Graham on the channel before. He does a lot of cool stuff with sygnathids and just cool stuff in general. But I'll let you tell us a little bit about yourself, Graham. First of all, what is an ichthyologist and what is it you do overall?
So an ichthyologist is someone who studies fish, and
Hmm.
basically it's the taxonomy and systematics of fish. So ichthyologists will examine species of seahorses collected from around the world, and it's up to them to determine whether they're new species or they need to be reclassified. And when I say reclassified, meaning sometimes the last century species may have been identified as so and so species, but it was done in error, and it's actually a new species or something else completely, or something well known. Um, and so I've been with the California Academy of Sciences and the Australian Museum since 2005, and I focus most of my research efforts on seahorses from the South Pacific, and I'm
Hmm.
slowly heading towards the Indian Ocean because that is the Wild West for seahorses and other species of sygnathids.
Sure. And yeah, the Indian. So are there not many species that have been discovered out there? Is that like all new territory?
This is the thing. Sometimes you'll have, like, for example, hippocampus hystrix found
Hmm.
all across the Indo-Pacific. But like with all other groups of fishes, they're now discovering, based on genetic data, that individuals of hystrix might represent a distinct lineage that occur in the Indian Ocean relative to those individuals found in the Pacific Ocean. And that's why
Gotcha.
I'm finding with certain other sygnathids that it's not just one new species found in India, but several.
Gotcha.
So yeah, so even hystrix is a new species of hystrix being described right now. I can't get into detail since I didn't write the paper,
Right. I
but
gotcha.
yeah, it's just the beginning. Yeah,
So, and what determines a new species? Because, like, I know, we got into detail last time you were on. When we talk about a species of seahorse, the way we say, okay, that this is this species, and it's because it's shaped like this, it has this many spines, it has
yeah,
this
yeah.
many whatever. Is that basically what it is? But you guys go into depth with using like,
Definitely, that's definitely part of it.
okay,
Um, Unfortunately, seahorses are morphologically conserved. They look alike across the board.
yes,
There's 46 species, 8 of which are pygmies, and that's a whole nother ball game. But with the
right,
non-pygmy seahorses, they're very similar looking. So I look at key diagnostic characters, characters that rarely distinguish between species. And for me, those characters are the absence or presence of certain spines on the head and body and tail. And it's kind of like a roadmap, and that guides you as to whether there are different species, and also counts if you know the location as well. It definitely,
sure.
yeah, yeah,
And so, for instance, the last time we talked, you had just been a huge part of describing the pygmy seahorse hippocampus japapigu, which
yeah,
is really cool.
yeah.
And you just mentioned that there may or may not be a new hystrix species that you're not describing, so you can't get into detail. But like, what goes into the description? Is that just writing the scientific paper, getting the proof, kind of thing? Is that how that works?
Yeah, it takes a lot to describe one fish species. one, there's the effort to collect it, which means you need to get funding to get to that country, or if you're lucky, someone in your field who's willing to collect it for you who lives in that country. So after Hippocampus japapigu, I was contacted by this young scuba diver from Sudwana Bay, South Africa, and that
Nice.
is pretty much the most southern extent of coral reef formation in the Western Indian Ocean. Then it
Okay.
gets cool, temperate. And she goes, by the way, I found some pygmy seahorses, and I freaked out because before now, this new species, Hippocampus nalu, it's the first time the pygmy seahorse has been found for all of Africa and extended the range by thousands of miles from its previous range from Western Pacific, namely Thailand, Cambodia, Australia, all the way to South Africa. It was a huge deal. So
Yeah, that's huge.
it's huge. So I said, I would love to get some specimens. I know some people in South Africa. Would you be willing to show them and help them collect them? And she eventually agreed. And,
I'm sure it took them a little convincing, but go on.
yeah. And then I had a team put together, and they went and collected a male and female.
Wow.
And so what you have to do, unfortunately, is you put them in ethanol to preserve their DNA and their
Sure.
body. But then once they're in the lab, I excise an eyeball from the right side. This is a tiny little piece of tissue. And then I do genetic examination of that. And what you do is you look at certain genes that act as barcodes. And once you have that barcode nuclear data, you compare that against a database of similar sequences from other seahorses. And right away, that genetic data told me it was totally different.
Wow.
So that was a good start. And then I examined it on the microscope, CT scanned it to look at its bones, completely different from japapigu and other pygmy seahorses.
Wow.
Yeah, so it took 2 years to describe it. That's what
Wow.
Yeah.
I mean, that's a long time, but it means that you guys are being extremely efficient, right? And making sure you know what you're saying and telling us right. And I mean, like, I know this is an out there question, but like, I mean, do you think there are just like so many more species that we just don't even know about yet? I mean, it's just about getting, like you said, getting access to them and then giving you guys the years to describe them. And wow. And
Yeah. I mean, at this point, currently there's 46 species of seahorses. I'm going to remove 3 from that list because they're not seahorses.
oh,
They're actually pygmy pipe horses, which I'm sure many of you know what they are.
yep.
They resemble seahorses. They're often mistaken for seahorses, but they're quite different genetically, and their bone structure is really different. And based on CT scanning, that really shows you what's happening. So right now I'm focused on reclassification of many species that were identified and classified as such many years ago, but in error. And so now I'm reclassifying these species, and it turns out it's more of an interesting story than we thought based on these reclassifications. So do you mind if I just continue along with this?
Yeah, no,
Okay.
please go ahead. Because I'm thinking of questions, but it's easier just to
ask
listen.
along the way.
Yeah.
So there's these 2 seahorse species, Hippocampus monitor and Hippocampus paradoxus. And they were found only in southern Australia, so they're temperate, cold water species. But what's unusual is they've only been collected from 300 feet or more. And there's only 8 specimens in Australian museums and nowhere else. Actually, no, there's actually 2 specimens in Australia, but there's just not enough of them to give us a picture of what they're like, what they do underwater. They've never been seen live.
Wow.
So I always thought they looked weird, but they look like seahorses at first glance. They're described as such, but within the context of further research is needed. So it turns out, based on genetic data, that they are closely allied with the Indo-Pacific pygmy pipe horses.
Oh, wow.
but they're not pygmy pipe horses. They represent an entirely new form of signathid.
Wow.
Yeah, they're not seahorses, they're not pipefish, they're not sea dragons, they're not pygmy pipe horses, nor are they large pipe horses.
Wow.
They're a completely distinct type or form or clade, genetic clade. And though they're sister or closely related to the pygmy pipe horses in the Pacific, they're completely different. So
That is so cool. And I mean,
yeah,
it's taking, because I mean, used to be, I didn't mean to cut you off. I will
no,
let
no,
you
not at
continue.
all.
But it used to be like back in the day, Holly and I, as hobbyists, we've talked about, how the books about keeping seahorses were so different because they were just doing what worked then and it worked. But
Yeah,
way back in the day, they're describing seahorses just based on mainly visual stuff. Like all the advances in science have allowed you to show that maybe not so much. They're different. That's so cool. Go on, go on.
yeah.
I feel
Well, I
like I
would
said
say
that.
with that, back then with those seahorses, 20 were created based on coloration, which is a big no-no.
Right.
And so I'm mopping up after that. So,
Gotcha.
but in a fun way. Yeah. Uh,
Right.
and that's how you learn what it constitutes a valid species. So that is really exciting to me. And so I'm working on this new class of signathid, but it turns out there is one specimen collected from South Australia near Port Lincoln, which is very great white sharky territory.
Okay.
One was collected at 80 feet. And it turns out it's a new species of this new clade of
Oh my
signathid.
gosh.
So not only do I describe a new species, I describe a new genus and also a new form of signathid all in one paper. So it's probably like the apex of my signathid research ever. It's a dream come true.
No kidding. And I
Yeah,
mean, oh my God.
yeah.
I'm so happy for you too. Like
Oh,
that's such
thanks.
an achievement for you. And it helped, it's so cool for all of, everyone, just to have you working on this. Gosh.
Yeah,
So there's
yeah.
this whole new species, and then there's like when Graham isn't, a scientist, so he starts
All
using
right.
words, and I'm like, so there's this new thing, and there's a bunch of new things underneath it.
Yeah, it's a new
Yay!
thing and a new thing and more of a new thing. It's amazing.
Right.
Yeah, it's
But it's still amazing. And I mean, and so, without getting too technical or giving away any of your trade
yeah, of
secrets or anything,
course.
like, so how does, I mean, can we say anything else about how the DNA is like, how do you tell that it's different? Or is that too
So
technical?
when you not really you extract tissue from a specimen
Right.
and then you have minute quantities of DNA. You need to produce a lot more copies to work with it. So it's a technique called PCR, polymerase chain reaction. The end result is you get lots of DNA. But then you have to narrow in one tiny area of that DNA, and it's a gene that's encoded within the mitochondria, and those nucleotide sequences from that little gene are used as a barcode. So you
Right.
compare the DNA sequences against other DNA sequences previously sequenced from other specimens. And so right away, when I lined those sequences together, I saw right away a pattern like, it's not falling with the seahorses. It's obvious,
Right.
but I can see it's in a group containing the pygmy pipe horses. But
Mhm.
then I thought it was a pygmy pipe horse. I'm like, yeah,
Right.
morphologically, when you look at the skeleton, they share some elements. But the more I study them, I'm like, nah. If you told me a spaceship crash landed on Earth and you scanned the alien, I would say it's that different.
Gotcha.
Yeah, yeah. It's
So cool.
yeah. It was a really fun eureka moment. Yeah,
Yeah, no kidding. And the more you expand and the more access you do get, I'm just curious how many true species we'll end up with. That'll be really interesting. Then you're like 46. Yeah. And like you said, he's taking three out of there because they
yeah,
weren't different
yeah.
species.
I'll be adding one more seahorse, which I'll talk about. It's
Okay.
it was previously valid, but then not valid. It's a weird, complicated taxonomic history, but I'm confirming its validity. So it's an
Okay.
actual true species from Western Australia.
Okay.
And Carey was instrumental in helping me with this.
And we're waiting on Chris still. He'll
Cool.
jump in. So
Oh, he's at the
go
pub.
on.
So on that, so remember we were talking about Hippocampus radii?
Yes.
How we thought it might be new species because Lucy had collected one specimen with a shorter snout than what's normally presented
Yep.
by the species.
Yep.
So I read this really cool paper that came out, and I always thought, I mean, I agree with it. There's this seahorse species, Hippocampus algirgus, from Western Africa,
Okay.
from north all the way to Namibia. And I always thought it looked like radii.
Okay.
When I CT scan individuals, specimens from both species, they look identical. The characters are very similar. Then
Right.
this genetic paper comes out saying, well, an offshoot of algirgus eventually made its way over to South America and then up to Florida.
Yep.
But genetically, they're very little divergence. They're almost identical. So I need more proof, but I'm working on merging those two species. Since algirgus was described first, that name takes precedence. So all these scuba divers will kill me, but they're gonna have to say algirgus and not radii.
Oh, so we would even have to then. If you truly got that done, it would be algirgus now and not radii. That's interesting.
Yeah, I mean it'll still be the long-snouted seahorse. I never know the common name, but
Sure.
scientific name. There's Chris. Is that Chris?
Yeah,
There's
that's
Chris.
Chris. He's trying to come back. And so real quick, I would like to throw this out there because as hobbyists, we've talked about this before too.
Yeah.
We all support seahorses in the wild for sure. We promote buying captive bred as hobbyists. Obviously, Graham was doing scientific work, different situation. But I would just say this lends to the theory that we've talked about repeatedly that there's a lot of times that they'll say that seahorses are becoming extinct because they're no longer in the place where they thought they should be or where they were before. And literally, this is lending to that truth that, oh my gosh, they may have literally
Move around.
really moved around in this case.
Yeah.
If radii is truly this species, then
Yeah, I mean, if I'm right, it'll be the first transatlantic species, but they don't mix. They're on separate parts of the coastline and ocean, but they just simply don't mix with the distances. It's a strange situation, but most likely they're the same species. But I need to show proof. And
sure,
one roadblock was getting specimens of algirgus from West Africa.
Sure.
So then I was just contacted by a group in Hong Kong whose government had an illegal seizure of seahorses. They nabbed these guys at the airport. Most of them were algirgus, getting 30 specimens to look at. So it's Christmas early for me.
Right. not for the one not for whomever was expecting to receive the seahorses, but yes.
Yeah, exactly. Yeah.
It still helps you, and that's a good thing. And so just I know this isn't even your field of study. This is just like a left field question. So if they are the same species and they're in different waters, different situations, I've always been like that. I keep saying it's my theory, and I'm not a scientist. It's just what I think. But if that happened, they traveled that distance over years and over time, and they likely, I'm not going to say, I know it takes a really long time for like adapting and stuff like that. But I'm just saying some things did change in what I don't know how to say it correctly, but could those two, if they're the same species, they still couldn't be like, if two were live collected, be kept together because they're in, you think they could? Okay.
Definitely.
Okay.
Oh yeah. Okay. I mean, there's been accounts of hybridization between two completely different species.
Sure.
Hippocampus rectus, Hippocampus radii. That's and then but yeah, so they theorized that these individuals or subpopulation from algirgus went over to the Western Atlantic 2 million years ago, which is long enough to create a new species in cygnathids. It's a bit too short. Maybe it was 3 million that were considered a new species, and most likely they're well on their way to becoming. But right now, most likely they're the same species.
That's so crazy.
Yeah, I
So
really have to
crazy.
know what I'm doing because otherwise I'll get dinged.
Yep. And well, I now that we've got Chris, just in case Chris gets technical difficulties again, why don't you go ahead and tell us about the species that he was so helpful with?
Oh yeah, actually, that's going to be. Oh yeah, so we're working on Hippocampus breviceps, which
Okay.
occurs throughout southern Australia, and previously it was considered one species from the state of Victoria to the state of Western Australia.
Okay.
But it has had a very complex history. Some people think the individuals in Western Australia were considered a different species called Hippocampus tuberculatus, and that
Oh
name
yeah.
was derived from its very spiny condition.
Okay.
And when you look at breviceps and tuberculatus, they look very, very similar. But so Carey was able to get me specimens of both species. And for the first time, we confirmed genetically that they are indeed two distinct lineages and two distinct species.
Oh wow.
Yeah.
Oh cool.
So what we're doing is we're resurrecting tuberculatus. So the distribution of breviceps now only occurs from Victoria westward to South Australia, whereas is restricted to Western Australia.
That's really cool. I mean, it's fantastic too because like literally in the last 5 minutes, you've told us that there is one species that's in two completely different areas where it's just a matter of they've traveled, and then there's two different species within a very close proximity, but they're different based on DNA and based on
Exactly. Yeah.
all of the things you use.
Yeah. And you brought up a good point, Kelly. It's interesting that you have two species of seahorses pretty much on the same coastline of Australia, but happen to be two. That's very unusual.
Yeah.
Yeah.
What do you think? I mean, like, just
Well, the reason
I mean,
the Great Australian Bight, it extends for hundreds of kilometers. I think even more, like 1,000. But it represents a really good barrier to having these two populations mix. And there's no good habitat. It's very turbulent, choppy, a lot of weather systems. So it has kept the two populations apart.
Yeah.
So it's been fun. So I had to get specimens from Western Australia and near Perth, and then look at lots of museum specimens, and then find out where they occur so I can collect them. And then Chris was amazing in getting me specimens from both species. It really helped. I mean, it was the definitive, specimens that helped me come to this conclusion. So Australia gets one new species, and whereas Florida loses one.
Right. It happens. And Chris, I know you have some questions too. I promise I'm going to let you. But I was just curious really quickly, Graham, how like when we were dealing with the seahorse with Lee, and we figured out that, not only do we know in the hobby, but you guys know in the ocean that hybridization within certain species, not all of them, not everywhere, but within certain is happening because the species are crossing over, and for whatever reason, they're breeding. So when a hybrid occurs, is that in your is that a new species, or is that just a sub? How is that to you, or is that something you don't touch yet because it's too?
Yeah, that's a really good question, and it's complicated.
Sure.
There's a very unusual seahorse found in this marine pond, and most of them look like Hippocampus erectus, which occurs from Canada down to Brazil.
Okay.
Or even down to Argentina. But they're smaller, but some of them have the head of erectus, some have the head of radii. So I think there was a past hybridization event that went on, and the morphology hasn't quite settled in a few of these individuals, but genetically they come out as Hippocampus erectus.
Okay.
If there's a lot of species of fish that have arisen because of hybridization, especially in pygmy angelfish in the Indo-Pacific.
Oh, sorry.
Yeah. But it's hard to say whether that happens in cygnathids.
Sure.
Yeah.
Well, I know that even like, I'm sorry, Chris, I turned you off for a second. I didn't mean to.
Okay.
I'm trying to let Cheryl
You
in. I
heard
can see
hybrid,
Cheryl's trying to get in.
I'm
Right. So, and I know like in the hobby, obviously I wasn't a part of it. I wasn't the breeder. I wasn't associated with the breeder, but at certain points they would sell erectus, but then there was this northern erectus that was smaller, shorter snout, different, so it just always interests me. And I wish there were more of you, or you had, more time to do this work, because, to get definite answers. Hey, this really is a different species or it's not, because I mean, it does matter. And it matters just in general to have the correct species. But then even when it trickles down to the hobbyist, it matters in how we keep them and such. I know that a question, I'm going to ask it for Cheryl because I can't get her in here. But I do know, I'm just curious if you know anything about this, and then I'm going to let Chris ask some questions because I know he's got some. But I know we were all fascinated with and wanted to learn more about Hippocampus zebra, right? And Chris, please correct me if I'm saying this correctly. I think there was some confusion, not sure about whether there was a Hippocampus zebra and a Hippocampus. What was the other one, Chris?
There's another
Was
one,
it
yeah.
Montebello winters.
Yeah. So
Montebello winter.
do you know anything about that, or is that something you haven't worked with?
Not much. It's something
Okay.
to be looked at later. Yeah.
Gotcha. Okay.
Yeah. I mean,
well, I know Cheryl wants you to.
Yeah, I can count 8 species that need to be looked at to verify whether they're valid species or they need to be comprised of these species. But yeah. Well, that'll be hopefully in a few years. We can talk about that. Yeah.
Yes. We're gonna, if we're still here, we're gonna keep begging you to come back.
Yeah.
And again, I want to say, guys, this is like a big deal. Graham is a true science, and Chris Carey's a big deal too. Everybody's a big deal, but I'm just saying he's taking time out to come talk to us, and this guy's really doing this stuff in the ocean. I mean, really studying. So it's really an honor that he comes and visits with us. We really appreciate it.
Yeah.
Chris, do you want to just start with questions, or am I putting you on the spot?
I'm pretty easy. Yeah, just happy to join in and have a chat with you guys. And yeah, sorry for all the technical difficulties on my end. The internet back at home isn't great, so I've gone for a bit of a drive and back on the road. And yeah, things are a bit better here. I can actually hear you guys, and yeah, you're coming through. But if it breaks up or whatever, then yeah, sorry.
No, you're fine. And I mean, my background always sucks too. I got my like teddy bears back here, so all good that we were in the vehicle. So I did want to ask, and you guys know, people at work tell me this all. I like jump to 5 different things. So if I'm jumping over something that you have a question about, just ask it, and I'll definitely get to talking about it. And as Graham gets to talking, we'll go back to things and such. But I know, Chris, one of the things that you had told me about when we were talking about having Graham on was the use of DNA. Help me remember exactly what the paper was about.
Yeah, so you can take this one if you want, Graham.
Are we talking about which species now?
Yeah, like Kelly's bringing up, like testing the water for traces of DNA of specific species.
Yeah, so that's called eDNA. So you detect strands of DNA that are just basically floating in the water column that has been basically leached out, released by the fish or whatever organism. And so the same technique I was talking about, amplifying quantities of DNA, it's a PCR technique. So you take lots and lots of water and you basically filter it through on a filter, and then you extract the DNA from that filter. And then you have these special techniques to amplify target species like seahorses, for example. And actually, with Hippocampus tuberculatus in Western Australia, my friend Martin de Brouwer, he and a group detected seahorse species using the eDNA techniques from waters off Perth, and they detected one sequence that didn't quite match, and that was the first molecular identification of tuberculosis ever made. It was a big hint of saying, yeah, there's something new here.
Whoa.
Yeah.
That is so cool.
That sounds so complicated because there's so many other DNA besides the seahorses in that water.
Oh,
So
there's hundreds of
how long does it take to separate it out?
Oh, not long, because the way you separate out is these DNA tags that are specific to a particular family of fishes.
Yeah.
Like for us to be the family Synathidae, you'll pick up anything within that family. So
Gotcha.
let's say I pick up 20 sequences from within that family using this technique, then I just have to compare it to existing online database. I'm like, oh yeah, that we know that when we know that one.
Yeah, here's
But then there's one that was different.
Wow.
And when I realized what it was, I said to Martin, let's apply for a permit to collect breviceps in Western Australia, which is really like,
Right.
it's like a fact-finding mission. I feel like, you know, the Hardy Boys, you know, just
Yeah.
try to resolve this mystery.
And what a cool job. I mean,
Very
truly,
cool.
just to be
Oh
able
my
to
God.
Exciting.
So putting that in like terms that someone like me can understand. Basically, when you're, because I would, you could show me DNA and I'd be like, you know, or tags or what, you know, all the things you're talking about. But basically, for a scientist who knows this such, you're saying that all sygnathids have these key things, and that's how you know it's a sygnathid. And then species have these key things and whatever they might be, and that's how you can tell, wait a minute, we don't have this one on file. Okay, gotcha. Really cool. And to take it out of the water, though, I mean, think how many things is that a new thing that they're doing, or have they been doing that for quite a while, extracting DNA from the water?
Probably 10 years at the most.
Wow.
Maybe a little more. But it's used more and more for all applications,
Sure.
especially for new species descriptions. But really because a lot of times when you want to, like, for example, there's a whole other group of ichthyologists study tiny gobies.
Sure.
And there's one in particular called Eviota. It's a genus that is very colorful, but very small, and they fuel the marine ecosystem because a lot of larger fish prey on these gobies. So they're the food of the coral reefs. And a lot of times they're just hard to find. You have to put a huge net over a coral bommy, add some, sleeping agents, and then the fish slowly go up and you collect them. But with eDNA, you can compare that to collecting and see what you're missing. And it turns out eDNA picks up a lot more species than what you're actually seeing in your net.
So cool.
Yeah.
Go ahead.
Oh yeah. That's it.
How far does it go down? This might be a silly question, but I'm just thinking about extracting DNA from the water. Does it go down? I mean, like, are you getting DNA of snails and stuff? Or, I mean, like, does it get the DNA of everything? Or
you can pick up anything you want, but it depends on the technique you use. Like those tags that are created specifically for, let's say, seahorses won't pick up anything else. It won't pick up snails or trout, whatever, And you can collect water from, down to a mile. It just depends where you want to collect.
Has that started proving that there are seahorses, deeper water species that we maybe don't know about, or is that something on the horizon?
No, that's true. But with sygnathids, any sygnathed that you find in the deep water doesn't mean it's a deep water species. It just happens down there because, like Hippocampus fisheri that you mostly see in Hawaii, it's been found at the surface. It's been found at 300 feet. So go figure. Yeah. So, I mean, there's many species collected down that depth, which are just common species found in the shallows. But I wish there were new species.
Yeah, right. Well, it's good that you have the techniques to tell whether they are or not instead of,
Definitely, yeah. I mean, when you look at these, every day, you get a sense of, okay, yeah, this definitely needs to be looked at further.
Yeah, Ray said he was born much too early because he would have loved to have done something as interesting as what you're doing. Absolutely. Hey, Professor Fish Tanks. Hey, KGU Aquatics. We're chatting with Graham Short, ichthyologist, who is in. He's our sygnathic guy for sure, but he's a scientist who does the work on the ocean. So any questions, spit them out for sure. Professor Fistank says, I remember learning about this in one of my classes. Yeah, it's really interesting, but I didn't learn about it in any of my classes. I'm old, guys. And the thing is, there's so much new stuff that they're using. I mean, that's Graham's whole thing is he's able to say, hey, this species you identified a long time ago, it's not a reed-eye. It's it's this other one. And so it's really good that he's cleaning it up and finding new ones.
Yes.
So when you're done with the project you're working on now, the cleanup and then the reclassification, what's next for you? Like how do you say, do these things just fall into your lap? Do Chris Carey's just come along and be like, hey, I think this is happening? Or like, what do you do when you're finally to the point where what you're working on is done? How do you find the next project?
I probably have 20 projects in the pipeline.
Gotcha.
Yeah.
I was say, I don't think he runs out.
No, right.
I need to calculate
Ray
my
decided to
lifespan
get into it tomorrow. I'm sorry. What was that, Graham? Sorry.
Based on my lifespan, what's left of it, I need to calculate how many projects I can sensibly do.
Gotcha. Yeah, I was gonna say.
Gotcha. I mean,
with us, it was actually the other way around. Like Graham actually came to me. That's how we actually met. I mean, I'm pretty sure, Kelly, you tried to introduce us back in the day, but I think it didn't work out because I think I was busy trying to breed reed-eye at Seahorse World at the time.
Exactly. Yeah.
And then, like, Graham had, like, absolutely perfect timing. Like, I had a bunch of samples of breviceps and tuberculatus that I was getting ready to actually send to scientists down in Hobart. And I was trying to get them to study this particular thing and tell me whether they were the same species or different. And basically, I wanted to know, what they were, whether they were the same or different. And basically, the scientists down in Hobart said that they would take the samples, but they didn't have anyone who was getting ready to look at them, or, they didn't have any intentions of studying them right now. They said that they might get to them 5 or 10 years down the line when someone wanted to come along and study them. And then right around that time, I got an email out of the blue, or I think it was a phone call. One of the staff members at Seahorse World took a phone call and basically said, hey, there's this guy who wants to talk to you about seahorses. And I'm like, okay, cool. And then, me and Graham started talking, and that's exactly what Graham was wanting to research. And I'm like, that's awesome. I'm going to put these seahorses on hold. I'm not going to send them down to Hobart. And then, we connected, and, we got him a whole bunch of samples. And, we've got him some samples since from some other places, like Ocean Reef Aquaculture has provided some breviceps samples as well. And
awesome.
That's exactly what happened. Yeah.
So Chris, I got a quick question for you. And sorry guys, I guess I don't get to take credit for this connection that they made. Darn it! But hey, I tried back in the day.
You tried,
Chris. So with the tuberculosis, if I'm saying that incorrectly, I apologize. And the breviceps, you've kept both. When you were working there, were they so similar? Like, why did you think they were different species?
Well, if you look at them, like, at the face of them, like, if you just take a brief look at both of them in tanks, you probably think that they're the exact same species, and that's why there's been, like, scientific debate for so many years. Because if you look at them on the face of them, they're both around the exact same size. Like, there's no real size discrepancy from one species to another, or no discernible one. There might be maybe a centimetre or so, give or take, from maximum size for them, but they're around the same size. They're very, very similar in, their shape. They're not wildly different in appearance. But if you're in a position where you're looking after them on a day-to-day basis, when you're looking after, you guys know, keeping seahorses at home, when you're looking after your seahorses on a daily basis, you're feeding them, you're looking after them, you're cleaning their tank, blah, blah, blah. you get to know the animals on a really, really fine level, and you get to know, individual seahorses from one another based on, their appearance, their size, just looking after them, you get very familiar with them. And keeping these two different samples, these ones that I know came from Melbourne and these ones that I know came from WA, kind of near Perth, I noticed that they had some differences on a morphological level. The breviceps seemed much more prevalent to or much more pronounced cirri growth. They were much more pronounced with all the cirri that would present on them. Not that that's a definitive indicator of them being separate species or anything, but more so it's the tuberculatus that have the bony tubercules or the bony growths up along their back and along their tail that are very, very pronounced, and that made me that was probably the biggest indicator that made me think these two seahorses, they're quite different, and then when you start breeding them and getting some fry coming through and that, and the fry are exactly like their parents, and there's similarities, yes, but the differences are quite pronounced. And I've encountered other people who've kept them over the years, and if you talk to collectors who've collected them for 20 or more years for the aquarium trade, everyone sort of if you call them a tuberculatus or a breviceps or whatever, everyone seems to fall on whatever they've heard them called in the past or whatever, and they seem to people get a little bit argumentative. It's just like, no, this is called this or this is called that, and it's like, I don't care what it's called, I just want to know, are they the same or are they different?
Sure. So real quick question for you is, obviously you kept both, you probably sold both, or either way you kept both. We'll just leave it there. Were they ever kept in the same tank? Did they breed with each other, or did they stay to their corners? That was you, Chris.
No, so I'm a big believer of not mixing species, but not even just mixing species. I'm very against, like, if you've got, say, a population of seahorses, and then you're getting in some other seahorses from a collector that have come from the wild, not mixing the two species, or like not mixing the ones that you've had in captivity for a good while, and not mixing them with more that have just come from the wild. There's problems where they might come with parasites, or they could release some sort of pathogen that might harm the captive populations or whatever. So I didn't mix them, and I had a suspicion, because they were from opposite sides of Australia, that they could be different. So I wouldn't mix them based on principle.
So cool that you got with Graham, though, and you proved your point. That's so awesome. Graham, so Ray was asking, he asked in our chat within Restream, what percentage of your projects do you never get to? But I'm going to expand that question to, how do you prioritize what the next project is?
Yeah.
You flip a coin? No.
Well, what I do so I live seasonally, 6 months or 5 months, 6 months in the US, 6 months in Australia. And while I'm here in the US, I am in the San Juan Islands, where I CT scan a lot of specimens to accumulate data. And when I'm in Australia, that's when I study that data. And that would lead me to what I need to do. Right now, I'm behind on 3 projects, one of which is the breviceps tuberculatus, and then 2 others. But I like to gather data when I think of it, and then I have it already, but I continue on with the older projects to finish them in the form of academic paper. Yeah.
I mean, I get it. My questions are all over the place, because my brain works same way, is, just make sure you get all the data, and then when you need it, you'll have it.
And you have it. Yeah.
Exactly. Yeah. I have all the data for breviceps tuberculatus. I have all the data for this really cool project I'll be working on. Hopefully, end of the year is I'm going to be splitting up the seahorses. So there's, to say whether they're still seahorses, it's going to be a really wonderful debate.
Wow.
Okay.
So I found, based on CT scanning, that the pygmy and non-pygmy seahorses are incredibly different,
I agree.
genetically and morphologically.
Yep.
I mean, and even though they're closely related, it's hard to say that the pygmies are actually seahorses. I mean, they are, but I would come up with a new name for them.
Mhm.
But then I
I
found,
like it.
yeah, I have one, but I'll wait till it's published, so it has an
It'll be a surprise. No,
Yeah,
I meant I like the idea too,
Yeah.
because
Yeah.
we had a guest on last week who is raising dwarf seahorses, which is amazing. And
Mhm.
he literally thought he thought they were pygmies.
I
And
hear that
so
a lot.
people get that confused, and we get up in arms. You can't keep pygmy, oh God, don't do that.
Yeah,
So them having a different classification would be nice for me anyways,
yeah,
and just it makes sense.
yeah.
Come
Well,
on.
Osprey will still be a typical seahorse. It's just
Right,
small, which we call dwarf. But a
right.
true pygmy is
Yes.
way smaller than Osprey.
Mhm.
No, right. And that's what I'm saying is I
Yeah,
think the pygmies should have their own.
correct.
They're
Yeah,
not really the
Yeah.
same in so many ways.
yeah,
And guys, by the way, just in case anybody watches later, when they're saying morphologically, they're talking about, guys, correct me if I'm wrong, but you're talking about shape, their attributes, the way they look, their spines,
exactly.
that kind of
Yeah,
stuff. And then genetically, we're talking about the DNA and what's like inside of them,
yeah,
kind of thing.
yeah.
Just to make that clear for anybody. We have some younger viewers sometimes. So, but what were you going to say, Graham? Go on.
And so even within the true pygmy seahorse group, there's a lot more diversity. So it,
Wow.
so it's not only I'll be separating the pygmy seahorses from hippocampus, the genus. I'll be creating new genera, which is the multiple of genus,
Mhm.
Wow.
within the pygmies, because the pygmies have been around for a long time. They live in highly specialized habitats,
Hmm.
and so they've changed accordingly over the years. But I am still the data gathering phase. I really have to be sure that I know what I'm talking about, because
Mhm.
lots of fellow ichthyologists are against the idea, and we have these spirited discussions.
Sure.
I mean,
I bet.
What's their side of the coin? Why do they want them to stay
because a lot of
hippocampus?
them are highly conservative. For example, um, I have a friend who works on clingfish. You find them on seagrass, on the rocks. Their girlfriends are modified to form a suction cup, and they're very cute. And there's a huge diversity of them in New Zealand and Australia, but they're found all over the world. And they look at it the old-fashioned way. You stain them to look at tissue, look at bone,
Hmm.
but no one's really ever stained a pygmy seahorse because there's just too few specimens available.
Sure.
There's plenty of non-pygmy seahorses. So they have to figure out on their own whether using their techniques, whether I'm right or not. And so I'm going to be collaborating with a couple. I said, listen, you know what? You can refute me. That's great. But I want you to show me that you can refute me. Because right
Yep.
now, based on what I'm seeing, I'm pretty sure I'm right. But it would be nice to have this recalcitrant group prove me correct or wrong. We'll see.
Yeah.
Yeah.
And how does that work? Let's say you get the proof that you need and you're sure of yourself and you write the paper, um, and they refute you. Like, does it go back and forth? Do you then, overcome
Really,
their
I mean,
objections or?
when you write an academic paper, it gets peer reviewed, and I can suggest which reviewers to have the actual experience to look at it, because it takes a jeweler to appreciate a jewel. They have to
Sure.
know what I'm talking about and
Yes.
appreciate where I'm going. So there's a few I have in mind, and
Hmm.
um, they will cross out everything that they don't like, add suggestions, comments. But after it's published, there's no back and forth unless it's
Gotcha.
on Facebook and there's some comments on it.
Sure. Sure.
Or they can actually publish a rebuttal.
Mhm.
Oh, okay. And
Yeah.
if someone publishes a rebuttal, what happens then? It just is.
It gets published in the same journal, and
Okay.
But you know what? I've seen this process go back and forth, and it's quite hilarious. It gets
Sure.
pretty personal.
Sure, I'm sure.
Yeah,
It sounds like Facebook for the higher class kind of thing, or the scientific.
yeah, for
Yes,
nerds. Yeah,
yeah. Well, I hope that doesn't happen. I hope you're able to cross all the T's that they need crossed,
and
and
that's
that's
why
part of your process, though, right? Yeah.
exactly I make sure I do all that and more. I tend to be quite verbose. I mean, I add everything I can to prove my point, show where I'm coming
Mhm.
from. Yeah, and
And go ahead.
go ahead. It's all good.
No, no, go ahead and
Yeah,
finish.
I was just saying, because I have access to the CT scanner here in the San Juan Islands, I can really show these characters quite large, so you can really readily see them
Hmm.
Yeah.
for comparison. True pygmies versus dwarf versus um, non-pygmies. And it gets
Sure.
more complicated because there is one species of non-pygmy, like Zosterae, but smaller than a true pygmy. So
Wow.
size is not the end all to say that's a pygmy.
Okay.
It's
Hmm.
really the bones inside the pygmies, the dwarf, the non-pygmy, and this tiny non-pygmy. So you have to
So
look.
wait a minute. So just so I make sure I heard you correct, there is a hippocampus, a
Yes,
seahorse,
yeah,
that you believe is part of the seahorse family, and it's smaller than pygmies, but at the same as the large. I always say
yeah.
adult. The
Structurally,
larger.
it's the same as the non-pygmies. All
Wow,
yeah,
Wow.
yeah,
that's
and it's just
crazy.
like the true pygmies. It has a restricted habitat, and it's bizarre and it's
I
stunning.
bet.
Yeah. So
Say, how do you even see them if they're smaller than that?
Right.
Is that one of the things you detect in the water, their DNA, and then go look for them?
rely on these wonderful dive guides who've been seeing them for a long time. They know where.
Wow.
Sure.
Yeah.
Unless they have
Yeah.
better vision than I do.
Oh,
Right.
yeah, probably not it. Yeah. So yeah. So there's more to the story. But
Yeah.
then there's
Please.
one species of seahorse, a non-pygmy seahorse, that um, has more in common with the true pygmies. So at every level, I'm discovering things that no one knew, and that I can't wait to publish.
No kidding.
And then I won't talk about it, but you'll find out when I publish that there are two pygmy seahorses, very well known, that have more in common with the non-pygmies, but they're still
Mm,
pygmies.
mm. Oh boy.
Don't blame me for having a migraine, but
No.
it's there's a lot of shared elements, but
Mhm.
the picture is way more complicated than we thought, because no one simply has looked at it. Everyone knew, yeah, pygmy seahorses are different from the non-pygmies, from the larger
Sure.
ones. But we externally, just based on size, you can tell they're quite different. But then when you look at them, at their skeletons, there's massive differences.
Sure.
Yeah. And that's just
Interesting.
massive difference number one. There's like 5 more massive differences.
Wow.
Yeah.
And with the skeleton, I know that you did talk about it a lot when in the hippocampus japapigu, but like, what do you mean by their skeletal differences? Like, is there anything else you can say to like
Sure.
tell us what you mean by
Yeah.
that? Go
Yeah.
ahead.
So I look at bones that form the skull. We say neurocranium, and the bones.
I'm
Uh,
still here. I just have to turn my camera off for 1 second. Go on, Graham.
so I am able to distinguish between species and between genera just based on these skull bones. And
Wow.
so based on the shape of those bones, I'm able to say, wow, not only are pygmy seahorses different from non-pygmies, but there's a lot of differences within the pygmy seahorses.
Between them, between
Yeah,
each other.
so there's
Wow, that's
more
complicated.
diversity than we thought. Yeah.
Very cool.
Yeah.
Very cool. Sorry, I
So
am still here,
how
guys.
many pygmies or that type of seahorse or similar to pygmy is there now that you've discovered that you
So the ones I've described, and I've described 2 species of pygmy, and there's another one in the pipeline. They belong to one group of pygmies.
Mhm.
So I found a few, and so each group will have their own genus.
Mhm.
So it won't be hippocampus anymore. It'll be something that
Mhm.
I'll need to create to describe these new groups of pygmies.
Mhm.
And there's only 8 species of pygmies. There's not that many. I think right now there's probably a few more yet to be discovered, for
I was
sure.
going to say, do you think based on this, based on the differences you found, that there's a lot more? That's kind of what
Yeah,
I'm
I know of
thinking
one.
based on what you said.
I know of one. But when I describe hippocampus nalu from South Africa, that's the most southwestern region of the Indian Ocean.
Mhm.
Yeah, there's still more. Indian Ocean is vast.
Wow.
I
So
love that name,
Well, there will
Nalu.
be lots of all types there then, sounds like, to be
yeah,
discovered.
Well, I mean, to
Very
be
cool.
real though, it's pretty ridiculous of us as people to think we've got it all figured out. The ocean's pretty
Right?
big. I mean,
Yeah.
and everything that Graham
No, you
is
see
figuring out
stuff
and finding.
every day, new
Yeah,
things they're finding in the ocean. It's so cool.
yeah.
I know,
I mean, there's basically
man.
3 species of fish described every year, but in the insect world, it's thousands.
Oh,
Sure,
yeah.
sure.
In fact,
We
there's
just have.
millions more yet to be described. It's just
Yep.
the manpower. Like, between Chris and I, we represent the main group of seahorse researchers in Australia. I mean, there are
Sure.
a couple more, but they do it more from an ecological aspect or angle. Um, I recognize that Chris, he knows the seahorses. He was able to distinguish between Breviceps and tuberculatus based on juvenile specimens right away. Because, a lot of times I just see these fish as dead specimens from museums, and
Right.
actually I get to see them in the wild. But most of the time I look at dead fish under a microscope.
Right. So
You
you're doing the genealogical part. He's doing the observation, seeing
Yeah,
that there's
yeah, very
differences, and
helpful. Yeah,
very cool. Oh, we appreciate Chris. We know what he contributes to
Yes.
Signethics for sure. We know how important he is. That's why we won't let him leave. We keep pulling him back no matter what. But
I like it.
so the one person that so do you work with Richard Smith at all?
I
Dr.
do.
Richard
In
Smith?
fact, he's been
Okay.
a co-author in many papers.
Awesome.
Yeah,
I
and he has an incredible wealth of knowledge because he's always out in the wild. So we work on pygmy pipe horses, and I've described one new genus from New Zealand. I'm describing another one from South Africa, but he's going to try to hopefully collect one from Fiji, and
wow.
then the Maldives.
Yeah.
And there's another one in Tanzania. So there's a lot more to be described. I'm not just working on seahorses, but there are pipefish species too. It goes on. So hence the 20 projects in the pipeline.
Right.
Yeah.
So what's the definition of a pipe horse? Like I know, but for any viewers, like what makes them different? Like, are they a combination of a seahorse and a pipefish, kind of
Not really.
ish?
They're similar to seahorses in that they have an advanced brew pouch to carry the young, just like seahorses do. They also have a prehensile tail, like
Hmm.
seahorses do. They A lot of them look like seahorses, but they're actually quite different. Once distinguish between two groups, it's quite apparent.
Yep. I was gonna try to go ahead. Go on.
So this new species of pygmy pipe horse from New Zealand, it was quite a surprise because there are 3 species of cold or temperate water species in southern Australia. The genus is Ichthyologist. And so the specimen found in New Zealand was thought to be a vagrant from Australia because they're next door neighbors. But when I looked under the microscope, I'm like, wow, this is completely different. It turned out not only the species was a new genus, and
Jeez.
so we called it Silex because it has a round cup-like bony structure on its head and has Greek for cup. And
Wow.
yeah, so then we found out later, there's a new representative of this genus in Fiji, the Maldives, Africa, 2 species, Seychelles, Reunion Island. So it goes on and on and on. And So right now there's 4 pygmy pipe horses, but I'm just fixated solely on silent. And I want to describe all of them as much as I
Sure.
can. Yeah.
So real quick for anybody watching, I'm going to share something real fast here, just to show you guys. First off, when in case, because I'm not good at, okay, so it's part of the family, but not the genus, and etc. So it's really easy to look up, guys. Just telling you where they fit in, and we usually start with the family. This, sygnathidae days and hippocampus. I can't even say that correctly.
Yeah.
All right, but it goes down to hippocampus. So I'm not good at that stuff, but so don't feel bad if you have to look it up, was the first thing. Second thing was, did this transfer over? Can you see the pictures now, guys?
Yep.
Okay, so this is a pipe horse. I mean, they're incredible. Like some of them look like pipefish, some of them, but it's just incredible that there's these species out here that we don't even know is enough about, and thank God for people like Graham. Was my point. And so kind of going again left field, I always do, but when you're doing a paper, you do all your collection. You said you do your collection this part of the year, then you go back to Australia and combine it and kind of put it into paper form and cross all the T's, etc. So is there ever a paper that just you write, or do you always have collaborators?
I always have collaborators. That's how I like to work.
Okay.
It comes back 100-fold, not only with making new friends,
Sure.
and especially in the fish world, you understand each other. For example, I have a virtual Signatha research group. One of whom is David Harasky, that Chris will know, but people from all over the world, and we help prop each other up in respect to giving talks, to finding specimens. They might know someone in another country that could help. If I could do it alone, but it wouldn't be nearly as fun, and I don't want to be greedy. I like to help people with their careers, because a lot of them are young career science, signed in early in their careers, where I'm, kind of in the latter half, and there's a new crop or generation of ichthyologists that I've come find here in San Juan Islands every summer, and they need projects, they need to learn, and
Sure.
so it's a blast teaching them.
That's awesome that you're teaching too. I would say I've not been paying attention to comments, guys. I'm sorry, but Professor Fish Tank said that's what I want to do is genetic work to finally give species names to aquatic life where there's no species names. Hey, become an ichthyologist. We'll hook you up with Graham. He'll teach you.
Where is Fish Tank located?
I'm sorry, what?
Where is he located?
Let us know, Professor Fish Tanks, because we'll see what he says. And while we're waiting on that, Zen Ginger wanted to know, you were never in the hobby, right? You didn't keep tanks. You went through college and became an ichthyologist.
No, when I was in 3rd grade, an unfair day, I won a goldfish by throwing a ping pong in a goldfish bowl, and that's how the whole thing started.
Really?
Yep.
Tell us the story. You threw the ping pong. Go ahead.
I grew up in Chicago with nasty winters. Eventually I had dozens and dozens of tanks in my basement, and I switched to guppies, and then to cichlids, which I fell in love with. And then when I was 32, I bought a boat in New Zealand and started this oceanic lifestyle, and that's when I started noticing weedy sea dragons in Australia, and that's when I became enamored with signathids.
Mm-hmm.
Yeah, and so it all started because of that goldfish.
That's awesome.
Yeah,
that's awesome. And that's the one place I will visit before I die. Dang it, is Australia. I already told
you're more than welcome to stay with me. Come visit.
And I'm going to. I'm calling all of you when I come, and I want to see everything. I can't imagine seeing the stuff, in the wild. Oh my God, seeing sea dragons
is amazing.
Oh my God. Okay, I'm there. That's it. Australian wine. All right.
Wine and seahorses. What more could you want?
Yeah.
Right? That's my dream. Oh my God. So Professor Fish Tank said New Jersey, USA.
New Jersey. And he's a marine biologist.
Oh, I'm
Okay.
so sorry. I'm missing some of the comments. I didn't realize you were actually a biologist. I apologize.
Yeah, he says, I'm a
Professor is
marine
truthfully professor.
biologist with a specialization in coral reef conservation, ecology, and aquarium sciences and aquaculture.
Bam, bam, bam. I love it. And thanks for joining us tonight.
Yeah.
See, we can totally hook you up with
Yeah.
Graham and with Chris for sure.
Study corals. Turns out a lot of coral species represent many, many species. So they're a cryptid species complex. So, that's a lot of people are into coral, new species descriptions of corals. Yeah.
Absolutely.
Yeah.
And it's scary too, because in the hobby they just kind of give them names, and that's not, it doesn't right.
Yeah.
Right.
Yeah.
So what was I gonna ask? I was gonna ask, let's see. Oh gosh, it left me, guys. Sorry. I had a great
That's all right.
question.
I've got a question then,
Yes,
if
please.
you're struggling.
Yes.
So earlier you talked to Graham about project prioritization, like how do you decide which project you're going to do next? I was just going to say, like, it'd be pretty opportunistic, wouldn't it? Like, basically, whenever an opportunity falls in your lap, like, you find someone like me who can provide samples with something, or you find another ichthyologist or another scientist who's wanting to study something oddly specific, and that you can collaborate and you can, or if there's funding become available to study something oddly specific, then that's a great opportunity for you to study it.
Precisely. That's exactly it. When we started chatting by email, then by phone, my interest went way up. And I realized, wow, I can actually pursue this project by acquiring samples that I can get DNA from. And that changed everything. I mean, we could have done it just based on morphology alone, but nowadays, if you include genetic data, that really seals the deal.
Yeah, it's got to be comprehensive enough to really sort of cover every aspect of it.
Exactly. Yeah. And it's great. All the data, and it's Southern Australia has amazing biodiversity for most fish groups, including cygnathids. I think it has more diversity than the tropics does. And
Sure.
yeah, cold water doesn't mean anything for diversity.
Sure. And we will put up Graham's. I should have put that in the post. I'm sorry. I will go back and edit that, and I will make sure that you can reach out to him on Facebook and whatnot. He does respond, absolutely. So
Okay.
yeah. But do you run into a lot of times? I know you said like some of these projects you're working on, getting the specimens was difficult because going through the hoops. Do you run into problems with I don't want to say customs, but just different countries and different places having laws and rules? Do you have to navigate that a lot? Is that
Definitely, it runs the gamut.
thing?
One is, in order to collect seahorses, you need a permit.
Sure.
It's basically an exemption of the laws preventing you from collecting. They give you an exemption to collect those. And, I've done most of my collecting in Australia and New Zealand and the South Pacific Islands. So I contact the local fisheries per state in Australia. But then sometimes I'm very lucky. I don't have to apply for a permit. I know the right people who already have a permit, who I know very well.
A Chris or whatever, right? Absolutely.
Well, like David Harassi, he's the main fisheries guy in New South Wales in Australia.
Oh,
and
yeah.
Like, as long as we collect together and we know what we want, it's all legal. It's great. We collaborate. But sometimes you have to jump through the hoops. You have people who are territorial, who want to prevent you.
Sure.
It runs the gamut. But eventually you do get the permit. And it may take years. Sometimes it takes a month.
Sure.
Yeah.
Sure. And Dr. David Harassi is amazing. I should have said professor in front of your name, Fish Tanks. I'm sorry. But Dr. David Harassi is amazing.
He is amazing. Yeah.
Some of his his website of pictures and images of seahorses is fantastic. I used it for a website that I did when I was in coding school, and I got his permission, of course. But I mean, he's really amazing. He works with the Seahorse hotels, and he's done a lot of stuff. for both sides, for the scientific side and for just seahorses in general. But just to be clear, so Graham, when you're talking about collecting and such, you just said, sometimes you hook up with somebody who's already doing it. It's great. And you can just get specimens, say from Chris. But a lot of times you're actually diving yourself, right? Like you're
definitely
going and getting them.
I prefer it because, I mean, I'd rather do it with, fellow friends and ichthyologists.
Sure.
But a lot of times I rely on the locals. They know exactly where they are.
Yeah.
And, I relied on one near Perth to get tuberculosis. I was told that they're rarely seen. In my 20 years of being in Western Australia, I've only seen one. Well, when I went on a night dive, I stopped counting at 50. So they're there. It's just you have to be in the know, and there's a lot of enthusiastic local scuba divers who know exactly where these fish are and
absolutely
contact. Yeah.
That's why I'm calling both of you
when
I come to Australia. Go ahead, Chris. Sorry.
Yeah, it's one of those things you have to know where to look, how to look for them, what to look for. In a lot of ways, you can't really beat that sort of local knowledge. Like, collectors are a fantastic resource for, the techniques and that they develop for how to look specifically for certain species. And, like some of the seahorse collectors, some of the aquarium collectors who collect seahorses in particular have developed fantastic techniques for finding them and, being able to collect them.
Well, yeah, you're talking about signathids that, I mean, their whole thing is camouflage. They're trying to hide from you.
Yeah.
I mean, they're not easy to just,
I can't find them in my tank sometimes.
Right.
Seahorses display nocturnal behavior during the day. Very difficult to find. At night, they're at the top of these algal bushes.
Yep.
And they're always there. Yeah.
So that's curious to me. You're saying they seem to stay at the top and
at night.
That's my tank. All my seahorses go to the top. They hang on the returns. They hitch on the Robert King char chamber, etc. They want to be. One of my males sleeps like he freaks me out every single day because he'll have his head, everything but his gills outside of the tank. Like his entire top of his head is out of the water. And like, this is like a total left field, dumb question, but I'm thinking fish, slime scale. Obviously, seahorses don't have that. They're not slimy. But wouldn't that be bad for them to be in the air like that? You're like,
not. He doesn't think so.
Right.
Likes it.
Yeah, apparently, right? So Professor Fish Tanks wants to know if you worked with the northern line seahorse. That's what I was saying earlier. We were talking about erectus, and they call this species. It's erectus, but they differentiate them in the hobby.
Northern and southern.
I don't know if there's a true differentiation. Do
there
you know?
There is a genetic divergence between the north and southern, or North and South America. But the difference is, so anything beyond 2%, you can say they're different species, but the genetic divergence for the northern and southern populations remain from 0 to 1. So maybe 4 million, 3 million years, they would be different species, but right now they're not.
Okay, can you say a little bit more about that? What did you mean by that? Because I understood, if it's only 1 to 2%, then it's not considered a different species. But it might be in a million years. Why? Because they change as they're.
The changes occur in the DNA over millions of years. They accrue mutations.
Okay.
When I say mutation, I don't mean anything bad. It's just a different DNA.
Yeah, different.
Sure.
Yeah. So.
Gotcha.
And that's the same thing with red-eye and Algyricus.
Oh gosh.
Yeah.
He's gonna take our red-eye away from us.
I know.
It's okay.
I don't care what they're called. They're still cool.
And am I saying it right? Algyricus?
Algyricus, yeah.
Algyricus.
Yeah.
That's a prettier name. So okay.
Yeah, I like that.
Yeah, it's not bad. But back to erectus.
Sorry, go on.
I'm working with Emily Rose who's currently assistant professor at Valdosta State University, which is part of University of Georgia. And she was just here in Friday Harbor in San Juan Islands, and we've been scanning Hippocampus erectus because there's something weird going on with it. It's the difference between male and females are more than what should be happening. And I don't know what it means yet. So that I was just looking at them before this meeting, and I want to know if there are any differences between the males and females in the bone structure. But I don't have much data yet. And this
Dang it, I've got like 500 questions now, but go on.
I know, yeah. I mean, I can't tell you much because I don't know much yet.
Sure.
We just scanned 20 of them, and so I'm cross-eyed, and now I have to process all this data to produce pretty pictures of them. Emily Rose is the Hippocampus erectus Zosteria guru. She's been studying them for years, and that's her focus.
You need to hook us up, Graham. She needs to come on the channel and talk to us.
You will love her. Yeah.
As long, I mean, if she's cool with me drinking wine and chatting like common people, we want her on.
She'd be perfect. Yeah.
See, I'm gonna message you. I want you to hook me up. But also, I know you can't answer any questions now, but as you go into this, that's really interesting to me because one of the things that we've talked about a lot as hobbyists is that when you're breeding Hippocampus erectus, until a certain age, obviously until a certain age, you don't know if they're male or female. But I personally, and Cheryl personally, who can't seem to get in, we've had situations where, in my situation, I had 2 males and a female. And they were the only 3 that I was able to, keep alive and grow through adolescence. And the female was so bullied by these 2 males, trying to score or whatever, mate, that she grew a pouch. And I really would love, I would love more information about the differences between males and females, and just, if she was just a late bloomer, or like if that was the case, why were the males trying to get with her so bad, right? And then if they really clownfish can change sex. It's just that'll be a fascinating topic once you get into it.
Horses don't change.
Okay.
So only those fish that school, whether it's large groups, the dominant male when it dies, the dominant female takes over and changes sex.
Okay.
I mean, I love to tell people who don't know much about nature that every single method to reproduce in nature is common, whereas male-female reproduction, we know it well, but it's not the best way to reproduce. Sometimes you self-clone, or some fish and lizards and sharks retain sperm their whole lives without needing another male.
Wow. Yeah.
Yeah. So males are.
But you're saying seahorses, they can't change.
No, they don't change sex. But
so if a female seems a female and she's a year old and she grows a pouch, she's just a late bloomer.
Totally bloomer. Yeah.
Wow. Okay.
So I wonder if they knew and they were bullying her because it was another male.
Maybe. Come on, Graham.
Who knows? I mean, you can tell, whether it's a male or female when it's older, and especially when you CT scan, there are certain bones specific to their gender. And it's near the slit where the eggs come out, or where the juveniles are born, but are released, not born. And with the erectus, there does seem to be differences between males and females that necessitate our looking into. It's something's weird going on, but it's not.
So what do you mean by weird? What do you mean like what do you?
No, it looks like the coronet looks different in males versus the females, but there's such great variation in seahorses, even among the same species, that I can't say for sure. Live yet. So that's why we scan them to see what is going on. Yeah.
Sure. And so when you do this studying of male versus female, will you, my only question is, are the bones different from, like, could you take a small, tiny 5-week-old seahorse and tell by a scan that it was male or female, or would you need to wait until the bones were fully formed and they were big? Or do you know?
I don't know, but I'm going to
Okay.
say you can based on CT.
Really?
Yeah, because once you're female, you're female. Same
That's awesome.
yeah.
Wow.
That's awesome.
Yeah.
Okay. All right. Because, yeah, that's, oh my gosh, that's going to be really
I know one
interesting.
difference I've noticed between my males and females is all my males have always been kind of stripy, like zebra stripes almost, and the females are always a solid. They don't get the stripes.
See, mine do, though. My males are, did you say the
Have you
females don't
had
get the stripes?
females? Yeah, I've never seen in my seahorses a female that
Yeah.
has the stripiness that the males do.
So I have,
Just an observation.
Yeah.
no, right. Yeah, I have, and that is something Graham said earlier, is lots of the species identification in the past was done based on like colors and
Hmm.
things like that, and probably not as efficient as, looking at bones. Good observation. I'm not saying, I
Hmm.
get what you're saying, but yeah, my little Cheryl had
Had the
stripes. So
stripes.
yeah.
Yeah, I just haven't seen one in the ones that I've had. Out of all the ones I've had, I haven't had any females get stripy.
Gotcha. I'm putting Graham's actual email in the chat there for you guys, for Professor and for whoever else. Just don't spam him, please, because we want people to come back. And once he gets this male-female research done, like, that's what's so cool is every time Graham comes back, it's something new. He's always doing something new and exciting. It's really, really cool.
Because we haven't talked about pipefish yet.
Yeah, hang on. And I do not
Or
want to
sea
keep you
dragons.
all night, guys.
Yeah,
Real quick,
It's a deep rabbit hole.
Zen
huge.
wanted to know, are there ever subdominant males that don't present as male as long as there's a dominant one already there?
I don't know. David Harashi would know.
Yeah.
Or perhaps even Chris.
I would question if maybe it could be like a development or a hormonal issue where they just don't present very, very strong levels of the hormone that sort of makes them go through like seahorse puberty, where they will develop their pouch. Like you do in aquaculture, I've seen it. You do get late bloomers where they develop those pouches, or they seem to develop and go through that puberty quite late compared to all their siblings. So I would question whether it could potentially be like an endocrine or like a hormonal thing where they're not developing the hormones to go through that transition through that life stage at the same rate as their siblings or brethren.
Interesting.
Yeah, that's a good point.
Yeah.
Or just related to that, when I look at museum specimens, I will see, like, for Hippocampus breviceps, similar size specimens will be at a completely different stage. Whether not only pregnant versus non-pregnant, but juvenile versus adult. I've seen a lot of juveniles the same size as small adults. So they're taking their cues at different times, different stages, but they're the same size. It's confusing at times. Yeah.
Sure.
Yeah.
Shyla said hippocampus. I'm not going to be able to pronounce that.
Hippocampus
Hippocampus
Yeah.
hema. Thank you. Has sexual dimorphism. Male has long. Thank you. Has longer tail and female has longer trunk. Okay, you're telling me something I didn't know. I didn't know that. So do we have time to, like, one of the things I never want to do is keep someone like Graham for like 3 hours. So we're not going to do that. Do we have time to chat about pipefish, or do we need to put it on next time?
No, sure. Let's do it.
Okay. All right.
Yeah.
Tell us about what's going on with pipefish.
God, where do I begin? So
usually the interviewer asks you a question and keeps going on. Just go.
Yeah. So it turns out that, like I told you about Hippocampus hystrix, individuals from the Indian Ocean are really a distinct species relative to those in the Western or Central Pacific. The same, you know the blue line pipefish eating aquariums, Doryramphus excisus.
Yep.
So there was a paper that prompted me to dig into this further that showed that Doryramphus from different regions of the Pacific, they don't mix. Genetically, they form their own group. So I was thinking, well, what does that mean? Are they their own distinct species? Does that mean that like there's hundreds of species across the Indo-Pacific, which would be amazing? Or are they just subspecies where they just don't mix at all? Anyway, it turns out I found out genetically and morphologically that yes, they are different species across the Indo-Pacific. So there may be well over 50 species of Doryramphus excisus across the Indo-Pacific.
Oh, wow.
Will represent the largest, what we call a cryptic species complex, meaning one species hides many behind the facade of one species across the Indo-Pacific. And I pretty much base this on, the bone again, the bone structure. And it's a huge project. I'm slowly gathering data, and that's why I've been scanning starting this morning as well. I'm scanning specimens from Sri Lanka and Mauritius to represent Western Indian, but also I'm probably CT scanning specimens from 50 different areas across the Indo-Pacific.
So they're saying, okay, this is one big, this, they're all the same, and they're not at all.
They look the same.
Okay.
But if you don't know what to look at, then they will definitely be the same, but they truly do differ. And not only genetically, like remember when I say anything past 2% divergence is a distinct species, well, we're talking up to 10% and beyond. So, and they look identical from South Africa to Mexico. And I've seen them live in Mexico, and that water gets cold in the winter. It's Northern Hemisphere, and or at least the latitude, it gets cold. It's not that farther south from LA. And based on the bones I look at, I can actually look at it as a picture taken from the wild by a photographer and tell you exactly where it's from.
Wow.
Yeah.
So, really cool.
Yeah, that's so cool. And the same thing goes with seahorses. A lot of seahorse species represent a cryptic species complex. And Hystrix is one of them. But and our Breviceps tuberculatus represents a two-species complex, which is kind of cool for Australia.
And I'm sorry, what was the cryptic species complex again? I'm sorry, I know you told us. I just missed it.
It's a complex of closely related species, or a group of closely related species that look identical, but genetically they differ, they diverge quite a bit. And a lot of times when they diverge genetically, you have to show, well, how are they different morphologically? Do they have an extra spine on their head, extra trunk ring on the tail? So you have to look at the nitty-gritty, at everything. And it turns out with the Breviceps and tuberculatus, it was really the presence or absence of spines, the angle of the coronet when they're juveniles. It's a combination of all these subtle characters.
Is there, I know you said 2% divergence when you're talking about that, but when you're talking about spines and such, is there a, like a number of things you have to have be different for it to be, like
it could
in your
be
consideration?
just one spine.
Okay.
Subtle as that.
Okay.
With Breviceps and tuberculatus, I can count 6 additional spines with the presence of those in tuberculatus versus Breviceps. And when people look at spines in a seahorse, it looks like a roadmap. It doesn't make sense, but there is a pattern.
So go ahead. Sorry.
Yeah, that's the key, is looking at the spines.
So now here's the real question. When are you going to take on the CUDA complex?
Oh, that's in the works.
Is it? Oh, I can't wait for that, because I know there's some stuff that needs to be done there for sure.
It's a really weird group. And when I mean CUDA, I'm including Algyrgus / radii, fisheri, and what are the others? I mean, there's like 3 more. So Borbanensis, CUDA, there's going to be a few revisions and a few additions.
Okay.
Yeah. And yeah, I've detected a lot of differences genetically. Now I have to show whether that is represented morphologically.
Gotcha.
It's an interesting group because this various seahorse species that comprise the CUDA complex, they split off immediately millions of years ago. So they don't diverge that much genetically. But in this case, when I say 2%, for this group, it could be 1% or even less. It's totally different.
Mm,
And
gotcha.
doing my head in because it's really different.
Sure.
This knows, you know, it's
That's why we need you, Graham,
yeah.
because you take on the headache of figuring it
I
out.
absorb the headaches for you, but I'm going to start slowly to chip away at this group by focusing first in radii algyricus because they're
Yep.
part of that complex,
Mm-hmm.
and then I can move over to like the others are distinct species. They are valid, but there are a couple like Borbiensis which have been merged with other species like CUDA, but they actually are a valid species. But
That's
genetically, I'm showing there's yet more undescribed species to that group.
what I think. I think you're going to
Yeah.
find more species that are not CUDA. Yeah. Yeah.
Correct. The original, so what we call a type locality is where was it originally collected that one could describe it as CUDA, and that was Malaysia. So I had a friend that's part of my virtual seahorse group online who collected a male and female near Singapore, and that is those 2 seahorses will be the reference. You compare all the CUDA found from Hawaii to Africa against the Malaysian specimens that he collected.
Mm-hmm.
Any differences I detect from any other population against those Malaysian ones will tell me a lot.
Yeah.
Yeah.
No, I'm sorry. I'm seeing things in my background, but no, I'm so glad you are working on it because that's one of the big debate discussions. You know,
it's huge, and
yeah.
But you know, a lot of time. I mean, the problem is when there's these online databases that have genetic data. The problem is, does that genetic data have a good identification associated with that specimen? Was that really CUDA that you sequenced?
To begin with, right?
Yeah, and a lot of times I would say 40% of what's in that genetic database, it's not good.
Yeah.
So you have to disentangle that information. So that's why I don't rely. I rely on the stuff I collect.
Right.
Yeah.
So we all just got to wait for Graham's papers just to figure it out. Sorry. Which is cool, and I get it. It's fine.
Next time we'll be in wheelchairs. We'll have a wheelchair wine meeting.
There you go. See, I love it. See, I mean, and it's really, you guys, this stuff is really cool to me because, you know, I was fortunate enough to meet people like Graham through different areas, but I mean, he's so cool, right? And he's so down to earth, and he's doing all these fantastic, crazy, really cool things, but he's not so haughty toddy that he can't come on.
No.
Yeah,
big egos in the sciences, but
awesome.
Yeah, I mean, we're all here to have fun and learn.
That's it.
You know, meeting you guys is wonderful, and you know, meeting Chris in Australia, we developed this total bond, you know, around fish, and we're propelling ourselves to do more. I mean, we have things in the pipeline that we won't mention yet, but it's the next few meetings we all have together. We'll share those.
Oh, can't wait.
Yeah.
And did I feel like I cut you off on pipefish? Did you share everything you wanted to say about pipefish?
So we talked about Doryramphus, and that's really the main pipefish group I'm working on.
Okay.
I'm scanning hundreds, and it's a lot of work.
Sure.
Other pipefish species, I'm working on some in Australia, and again, they're cryptic species, meaning the one species that we know and love really represents another 2 or more.
Yep.
Yeah, and I can count 3 right now. Based on genetics, there's a very east-west distribution going on. You know, the typical temperate Indian Ocean in Western Australia versus New South Wales, Victoria. And those 2 species tend to mix in South Australia, you know, just like our Breviceps tuberculatus. So there's a general pattern to be found. Like if I'm in Australia, I'm like, well, I'm going to work on a new species, collect some in Eastern Australia, collect some in Western Australia, compare them. It's a good start. Yeah.
Gotcha.
I've got a question for you, Graham. So with with studying pipefish or even seahorses or any cygnethids that you study, what number of samples do you need to get a big enough subset of data to
good
like
question.
fundamentally describe a species, or tell the difference between 2 very closely related species? Like, how many samples would you need on both sides to properly scientifically describe almost,
You know what?
beyond reasonable doubt?
Correct. You know, sometimes you only have access to one specimen, so you do it based on that one specimen. Like with hippocampus, nalu, and japapigu, I only had 2, but fortunately they were both male and female. But because I'm working with bone and not soft tissue, you know, bone is genetic and it's not that variable. It's there or not, including its shape. So I'm able to describe new species based on 2. But with pipefish, you can definitely collect a lot more. So I would at least 6 up to 10 to show that the pattern holds, you know, genetically.
Yep. And that's for each location variant, like from each, island or each river or each
like
wherever they come
from? With pipefish, I'd want to collect from each state. But I would most likely, generally there's a pattern from New South Wales, Victoria, they're generally the same species. But when you get into South Australia, and particularly in WA, most likely it'd be a different species. But yeah, I always say 6 or more. And we're lucky with Breviceps tuberculatus, we have more than 6 per species. And pipefish tend to be way more abundant. And logistically and legally, it's easier to collect a lot more numbers of pipefish. Yeah. That's a very good question. I mean, when I described japapigu, I had a reviewer on the paper said, "Why don't you have hundreds?" I'm like, "Well, because you don't find hundreds of pygmy seahorses. You find a few if you're lucky." Yeah.
Yep.
I mean,
no.
Yeah.
Go ahead.
Yeah.
Go ahead.
I was going to say that I know plenty of ichthyologists who describe new species based on one dead specimen from a museum that was collected 100 years ago. It's enough. Yeah, and you can acknowledge that, saying, "Yeah, I wish I had more, but I don't dive to 300 feet. I can't get it."
Sure.
Yeah.
But no, what you said makes sense. The bone's either there or not.
Yeah.
I mean, it's just, there's no real question. It's just when you get into like 2% divergence and there, how many things have to be different for it to be a species, it matters. But
like in all humans, the skull shape is the same. Our next closest relative is the chimpanzee, and there's 2 species of chimpanzee. Well, we want to compare our skull to a chimpanzee, and that's where you learn the differences between the 2 groups. So
we are different for sure.
Well, except for some people, but
right.
Yeah.
Oh gosh. See,
I've got another question for you, Graham.
Yeah, please go ahead.
So how important is it to you to know the origin of the sample? So what I'm asking is, it's one thing for you to go and collect a sample of a seahorse or a pipefish or whatever from a specific, a reef somewhere or a specific location. But if you get handed a sample that you get told came from the aquarium trade, so say someone bought it at an aquarium store, and they don't know the origin of it. They don't even perhaps know which country it's come from. Having a country is a good start, but a country as vast as Australia isn't really that good of information to know, not even what state or what area specifically it's come from. How I guess valuable or how good is the data that you get from a sample that comes from a nebulous source? Like, you don't know the origin of where it's come from.
It's really important to know the origin. You know, including the lat longs, everything. We have used specimens from unknown origins. We at the very least know the species, but we have to say the origin or location is unknown. But it really helps, especially with seahorses and pipefish, because a lot of them look so similar. And if I'm looking on Dory ramphus, I have to know the location, otherwise it's useless, unless that morphology of it conforms to what I know already from other populations. But it's, I would say 100% you need to know. Yeah, that's a good point.
Yeah.
Yeah.
Because I was just thinking, like, you know, it's relatively easy to get samples within the aquarium trade. You know, seahorses come up, and, you know, pipefish come up in the aquarium trade. So that could be a valuable, easy source of seahorses or pipefish for samples to help build up a decent data set. But how good is it if the data source or the source or origin of them is sort of nebulous, like you don't know exactly where they've originated?
It's not good. I mean, sometimes it's useful. Yeah, I know the species, I can work with that. But if you want to know, if you're doing genetic work and all of a sudden there's two species that are very similar but differ genetically, and I don't know the location of one, then it's useless to me. And it's a sad state of affairs, because I could have written the paper on it, but I can't. So that's why I rarely buy from fish stores. I mean, Vermiceps would be different because I know the origin based on the collector's information. But if I were to buy radii, well, I need to know whether it's from South America, or most likely it's from North America, but it'd be good to know. Yeah.
Yeah. I mean, the reason why I asked is in the cases where I've given you samples, whenever I've sought samples from a collector, I ask the nitty gritty. I say, you know, where exactly has it come from? You know, because, like, I just guess I'm a little bit OCD and I have to know exactly where it's come from.
Yeah.
But quite often you don't get that information.
KJE Aquatics said, for many fish, the collection points are very important, even in the hobbyist realm, because
they want to know now.
Yeah, exactly. People care now, and especially when you're writing a paper, though, too. You know, you got to make sure, like the situation with Lucy collecting a hybrid. You know, I mean, that's not the same, but I'm just saying the collection point matters. I totally agree. And I was trying to go to what CoralWorks said.
Yeah, Coral's asking whether there's fossil records of seahorse, and there's many from Italy and Slovenia.
Oh.
Yeah.
Cool.
Go figure. But yeah. And there's pipefish fossils from Slovenia, from California. But yeah, this fossils for most major groups of cyngathids. Yeah.
Gotcha.
Yeah.
All right.
And just going back quickly to the the seahorse sample thing. I was also going to say, like, it's probably a sad source to bring up, but there is also the traditional Chinese medicine trade. That's an easy way to get samples, but again, you've got no idea where they've come from, because, you know, a lot of the collectors who collect and dry out the seahorses for that trade pillage all over, and, you know, it's on the black market, so you're not going to get the information to go along with it.
Right. Each year I'm contacted by certain governments or departments that have confiscated legally collected seahorses. And this year is from the UK and from Hong Kong. And we're talking about hippocampus cuda. I received a huge stash of cuda and other East African species. But when I say East African, it could cover a huge area, you know, from Madagascar, Seychelles, up to Kenya. I know what they are, at least superficially. but it would help if I knew the country. It's just that extra data really helps. And then when I was talking about Algiricus, I got a huge stash from Hong Kong, but they told me the seizure was from Senegal, so that actually helps.
Mhm.
You know,
But wait, I am confused. So I understood Chris's question, and I understood. I think I understood everything you said. But like again, going back to if they have specific markings that when we're talking DNA, that tell you what genus and all that
yeah,
they're
genus,
from,
species, yeah.
why would knowing the location matter? Am I confusing myself here? Like
No,
I understood Chris's question, but
yeah.
then if you get
It
down into it,
matters
you have the
because
sample.
let's say I have CUDA from South Africa,
Okay.
but then I have a CUDA from Southeast
Okay.
Asia, like
Yeah.
Malaysia. I would really have to know because if they differ genetically, and they do,
Right. I get it.
if I don't know the source of the Malaysian one, I would I would down a whole bottle of wine or two
Yeah.
because
Right.
I can't finish the product or
Right.
the project.
Got it. Got
So
it.
yeah, it's important to know the location so you can say, well, hippocampus CUDA does differ between South Africa and Malaysia,
Right.
and I can say definitively, like, yeah, and Malaysia was where they were originally collected. And I want to acknowledge, is it Shyla?
Oh,
Mm-hmm.
Yep.
China in
what'd you say?
Singapore.
Yeah, I just want to acknowledge that. Yeah, they do have CUDA and Comes there.
Mm-hmm.
Tiger seahorse. Yep.
Yeah.
Yeah.
And my collaborator there, his name is Adam Lim, and he would be another great person to have a meeting with. He is hilarious, and he knows a lot about seahorses throughout Malaysia, and fun guy. Yeah,
So I'm gonna make Graham hook me up with all these people, you guys. But
yeah,
what I'm saying is would be really cool is if we could somehow get everybody in one. Oh, that'd
yeah.
be so
I'll have
fun.
everyone I
Oh my
know,
god. Yeah,
and
be
we'll be
so
drunk
fun.
by then. I mean, honestly,
Yeah.
at the end, we'll be drunk. So,
That's fine by me. Fine by me. But so we're running on 2 hours, guys. I know that I haven't asked all the questions. I know we want to know more from Graham. I'm not trying to pull at him because we got to give him time to do his research and get the papers written before he, tells us something and then, he
yeah.
tells us a secret or something. He's not drinking wine tonight, so we're pretty safe there. But
Oh, darn.
I'm kidding. But
I know. I'm so
Might be more fun if
surprised.
we give him some
next
wine.
time we'll have him
Yeah.
drink wine and we'll find out what's coming up next.
Yeah.
Really. But
I do have one question for everybody.
yes, please.
I need to find specimens fresh of radii. And
Okay.
I recently came back from Florida, and in the areas where we would have been legally able to collect, we didn't find any. And I'm a little reticent in getting some from the aquarium industry unless they definitely know where they're originally collected or bred, after collection.
Sure.
So just want to put that out there if someone knows where I can get some.
I think I might be able to help you there. I think I might know someone who might know someone who could help you there.
Okay, so we'll talk more about that. That'd be great.
And I think that Chris would be the best alternative because we don't have, like as hobbyists and on our side of the spectrum, we don't have these species anymore, unfortunately.
Yeah, radii is hard to find.
And I don't think it's because they're going extinct or any of that stuff. I just think that the industry has found it less expensive.
Yeah.
Yeah.
Difficult to
But
breed
it's
them.
interesting that when you went to try to collect, there weren't any. That's really interesting. Hopefully they just moved. But
Yeah, they're there. It's just
yeah,
limited time.
Finding them.
finding
Yeah.
them. Yeah.
And they're really hard to find in Florida. I mean, they've been collected in deep waters in the Gulf of Mexico by the hundreds, and that's based on museum specimens, which is surprising that
That's
they're
so
found in
crazy.
shallows and deep waters just like that. But simply flying to Florida, east or west coast, it's harder than you think. I mean, Zostre is
Sure.
easy. Yeah. And
So is Ostre just easy because they stay in the lagoon area?
yeah,
They
and plus,
stay up
because of
top?
Emily Rose, I know exactly where they're found.
Yep. Yeah.
I also know where they're found on the east coast of Florida, but I'm not able to collect there because they're popular dive spots. You know, I'll never want to collect from there. It's a big no-no. But
Gotcha.
in areas that are not, you know, touristy, then, you know, it's good to at least try. But I couldn't find any. And I generally just
Gotcha.
need two. You know, I'm not there to crash the
Sure.
competition. It's just
Mhm.
a couple.
Right. I think we've there's no case to be made, but I think it's pretty obvious that what Graham does is really important. And you know, actually, Graham, why is it important that we know the species? Why is it important for this differentiation and knowing, you know, the
Yeah.
difference? Why is it important?
It's all about biodiversity and conservation. The more
Okay.
we know what's in our backyard and around the world, then we're able to say, well, that's a new species, or that older species is now restricted to this location. We need to conserve them better, and then enact policies to conserve them. And
Absolutely.
basically, in a nutshell, yeah,
No, and that is a nutshell because it goes much deeper, but
yeah,
everything he's doing is really important, guys. So, you know, a pair of seahorses is not, you know, is very important to what he's doing, is my point.
true,
That's all I was
yeah.
trying to
And
say.
these fish collections at museums, they represent a snapshot over different decades and eras,
Hmm.
and they're a great resource. Because
Yeah,
in a couple of years, this world will be very different. And so
sure.
those future generations will be able to look at fish species that used to be here in different numbers. So museums,
Yeah, in 200 years, guys, it won't be me, obviously, running Wine Wednesday, but whoever is will have someone on that'll be saying, oh, that Graham Short, he didn't have enough science. We have all this equipment now, and I've scrapped everything he did. No, I'm
yeah,
just kidding. I'm
exactly.
just kidding. I'm
Yeah,
just kidding.
I'm writing a
But
rebuttal 200 years later.
right, the further we go, the better we understand. And
Yeah,
understanding is key to so many aspects of all of this, whether it be conservation, whether it be our hobby, whether it be any of it.
yeah,
It's understanding what we're dealing with. Like, you know, even coming into the trade, that's kind of what I was going to ask you earlier, Chris, is, you know, is the care of both, now I'm going to forget their names. Of course, I have been drinking wine, but what were the breviceps and the tuberculosis? Is that the two, or am I mixing it up? Okay,
Yep.
so are they? If you sold them, are their care the same for a hobbyist?
Uh, Similar, but they might differ a little bit because like where they're found, the
Temperature.
location's important from the standpoint that it'll give you a bit of an indicator as to what temperatures they can handle
Yeah.
in captivity. You know, in keeping you know, any animal in aquaria, we're very lucky in that we have control over, you know, what temperatures we can make those tanks go using heaters and chillers. But I mean, really, what we're trying to do at the end of the day is replicate these animals' natural environment as closely as possible. You know, whether we're doing that all that well, usually we're probably not. But at least if we can give them their basic needs, you know, if we can give them habitat where they're safe. I mean, it doesn't really matter how close it is to their natural environment, but if we can replicate at least the water chemistry and the water
Temperature.
parameters of their natural environment as closely as possible, that's a really good start to keeping them successfully long term in the hobby.
No, agreed 100%. You just answered it perfectly the way I wanted you to.
yeah,
Thank you
really
so
good.
much. But I mean, it just matters in every aspect. Maybe not as much
Definitely.
to us,
Yeah.
but you know, in the scientific community, it's really important. So we
Well,
appreciate. Graham, did you have? Go ahead, Chris. Go
Oh
ahead.
yeah, I just want
sorry,
to. Oh, sorry,
you
Shiloh
go,
just
Graham.
mentioned Adam Lim. He's considered the seahorse whisperer in Malaysia.
Okay,
That's really cool.
We
The
definitely need him
Malaysian
on board for two whisperers.
Shyla, I'm gonna do my best to get all these people in one Wine Wednesday with, you know, different times. But you make it, so I don't see why he couldn't. But we will try to get all these people. But
You would love it.
man,
Yeah,
yeah, reach out for sure. And as you can see, people like Graham are very approachable. They're so cool, and you know, they blow us away with their knowledge, but they're so down to earth that, you know, they'll talk to us, right? That's I mean, that sounds ridiculous, but it's just nice that, you know, we can learn from you and that you're willing to come and chat with us. I
I'll even
really
drink with you,
appreciate
I promise.
it. So Graham, did you have anything else? And then I think Chris has one more question.
No, that's it. I thoroughly enjoyed tonight. Thank you. I really.
All right. Please believe we'll be bugging you for answers on all
Please
the
do.
maybes and almosts, and I'm
Yeah.
working on this next year. We will have him back on again. I promise
Done.
you guys.
I love
And
that.
Graham, you're
Awesome.
welcome any week, of course, that you're
Thank you.
bored. Jump on in, but
Thank you.
we'll have you as a guest to actually talk about the specifics when you're ready.
Okay.
But Chris, what was your last question?
The thing I was going to say is the reason why Graham's research is so important is because what's changing fundamentally isn't the number of seahorses that there actually are. That's not really changing. It's pretty relatively static. It's our knowledge of what's out there, of the different species and how they're different. Our knowledge and understanding of seahorses and their relatives is fundamentally changing as we go. And, you know, the more we learn, the more we realize that we didn't know from the start. Like, you know, just the differences between these groups of seemingly closely related seahorses and the differences and little subtleties between them is fundamentally changing. But like from someone who, you know, I'm deeply embedded in the aquarium trade, and I guess my interest in all this is that I would love to, you know, introduce more species and more, you know, varieties of seahorses for the aquarium trade. But you know, when you're taking a seahorse species that you know next to nothing about, a lot of our species of seahorses here in Australia are so data deficient. You know, aside from abdominalis and whitey eye, most of them, you know, when Graham approached me, you know, the problem was that we didn't have any information or data. The scientists haven't studied them properly to even know what they are and what species is what.
Amen.
So, you know, from my standpoint, I want someone like Graham. I want the Grahams of the world to come and, you know, study our seahorses. And, you know, Australia is a massive biodiversity hotspot for seahorses and cygnathids. So, you know, I think it's about time someone like Graham comes here and starts doing the research on all of our species to fundamentally, you know, leap forward our understanding of all these beautiful animals. And, you know, if you're trying to take a species and bring them to the aquarium trade that's never been done before, having a greater understanding of exactly where they come from, their ecology, their natural life cycle, you know, the little subtleties in their development, you know, basically understanding their morphology and everything all comes into play. And you know, Graham researching and understanding that, and basically, you know, writing his papers and helping everyone across the world understand this about these species is just so critical to that.
Absolutely. And I wanted to say too, I'm sitting here saying how amazing Graham is. I want to say too, Chris is really amazing too. We're honored to have Chris here too, because, you know, he literally helped Graham make so many things possible. He's done so much for the industry and for the hobby. Chris, we appreciate you too. I'm sorry, I'm tooting Graham's horn and I forgot you.
No,
sorry.
No, Chris is a great guy, and I love our, you know, just how we collaborate. You know, we get in this
working relationship. Yeah, absolutely.
Down this fun rabbit hole. You know, I love it.
I know. I hear listening. That's why I had you both in, and you didn't do it, and I'm glad because we'd be here for 5 hours. But listening to you two talk could be. I mean, you guys don't even know. They could go back and forth all night, and me and Holly would just be like, "Really?
Yeah,
wow." You know, I mean, like,
yeah.
But it's fascinating, and we appreciate you both and all the work you both do. Zen, I saw your question. I will answer it. I do want to, you know, let Graham go because we've kept him for 2 hours now. But go ahead. Did you have something else to say, Graham?
No, I'm just enjoying.
Okay. All right. I mean, you're more than welcome to stay, but I won't question you anymore until the next time you come, because Zen had a hobbyist question. But I did want to say too, real quick, for anybody watching, last week we had Aqua Balls on. Love the name. You have to understand the meaning behind it, though. And he gave us a tour of his facility after I stopped being live and I was like smacking myself. Why didn't I make him do it live? We will have him back on possibly next week. I'll work it out with him when he's going to come back, but I want you guys to see the tour of his facility. It's amazing. All right.
Cool.
Then, and again, Graham, I'm going to answer Zen's question, but if you have to go, we just thank you so much for being here
Thank you guys.
and sharing
Thank
with us.
you.
Thanks so much. Great seeing you, Kelly.
Yeah, we'll have you
Nice
back
to meet
on.
Holly.
Great
Yeah.
to
All
meet you. That was awesome.
Bye,
I know.
everyone.
So cool.
Take
Have
care.
a good one.
Bye.
All right. Because he doesn't care about our tanks. I'm just kidding. He absolutely cares about our tanks. He does. But Zen wanted to know, and I did forget to say, Shyla, I just understood now why he was saying that Adam's the Seahorse Whisperer. I'm totally down with that. Like I'm to the point where we're all Seahorse Whisperers. On the website, that's what it says. Ready to become a Seahorse Whisperer. It's not my name. It's our name, right? You are too. So anybody who cares about Seahorse Whisperers and Zen said
Chris
I have
has
5
a cool
hours to.
shirt.
Oh, sorry, Chris. Let me make you big.
Look at that shirt. Is that cool?
Wrong
That
color.
is cool.
Yeah.
We gotta get. Listen, guys. I'm going to Holly's in what, a week and a half? It's really close.
It's really close.
I'm going to Holly's on like the 7th, I think. I can't even remember my own plane.
Yeah.
But I am going to Holly's, and we are going to hash this out and get the shirts going for Seahorse Whisperer. So, Chris, I'll send you one because you're a special guest. You get one for free. Sorry, guys. But Zen did have a question, and I was hoping to try to get Cheryl in. I just couldn't get her in. What's the one thing a new seahorse keeper should keep in mind setting up? I have my answer. What do you say, Ray?
Just realize you don't know anything.
You're an idiot. No, that's not it.
No, it's not that. Just I went into
There's
it
always
thinking,
more to learn.
yeah. When I went into it, I thought I knew enough to be successful. And it took me a long time to realize that I was totally wrong.
Well, and real quick, before I move on to the next person, Ray, it was interesting. I actually, because I went on Facebook, I'm not doing that often enough, for sure, guys. I'm sorry. I need to post my new seahorses. You guys haven't even seen my new seahorses. But I know I'm going to try right after we're done. But I was going to say, I saw a post in the Seahorse Whisperer group, and it was about setting up a new tank. And I saw some things immediately that I wanted to say, but it like would have taken too much time to write it out. So I'm going to go back and do it for sure. But do you find, Ray, that someone that's had experience with reefing or say freshwater, like, is it so different that they have to, like, does that play any part into what you say about, knowing what you know before you go in? I know I just rambled. I'm not asking.
I think the biggest problem is there are too many variables and you don't know. So just assume that nobody knows anything.
Okay.
And do the best you can with that. Unfortunately, I'm a person that whether I'm talking or whether I'm writing, I can't do it in just a few words. So
Sure.
my postings tend to be very long.
Mine too. That's why I couldn't do it. Go on.
Well, I go ahead and I do it anyway. And I suspect most people start into it and then sort of drop out.
I don't think so, Ray. I think they read them. I think it matters. I think you matter for sure.
Yeah.
I like them. They're comprehensive.
Yeah, I do. Yeah, I
Me
read it
too.
all.
Chris, what are your thoughts? The one
On
thing
the one thing, I think there's so many things. Picking one's really, really tough. But if I had to choose just one, I'd say like keep it simple. Just keep it down to keeping good water quality and good food.
Okay. And what do you mean by good food?
Good food that has all the nutrition that the seahorses need. So like good quality mysid. That's fresh. I mean, obviously most people go with frozen, but fresh frozen, if that makes sense. Like not really, really old, been in the bottom of the freezer for 6-12 months. really good quality food. And that extends to Artemia and your copepods and your live foods and that as well for younger seahorses where that's applicable. But fundamentally, just good food, good clean water, and keep on top of your maintenance.
Holly?
Well, after thinking about it, I had time because there's so much to it.
Yes.
I'll come in and say some of the things I wish that I had known and planned for was knowing ahead of time what you're going to need to take care of them and what kind of costs, your budget, because you're going to need salt for their water changes, their food, their equipment. So, and also your time, like if you want to raise the fry, look into that first on what it takes to raise fry before jumping into it to see that you'll actually be able to do it with the lifestyle that you have. So I would just say, don't just jump into it. Don't be the person that goes to the pet store and comes home with a seahorse and then looks up what to do. Find out first and plan. And I mean, coming from Zen, she's been around long enough. I think you got it. Honestly,
Mm-hmm.
I think you'll be fine. You know
That's
enough
it.
to get started. If someone came blindly and said, I bought a pair of seahorses today, what do I do?
Right.
That's a little different. But yeah,
All
I would
right.
say for the biggest thing for like somebody who has no clue and is just thinking about, hey, I want a seahorse, I'd say the biggest thing is research what it takes to keep them before you jump into it.
So Zen said, what I'm hearing is, even though I've researched for almost 2 years now, I think I know what I'm doing. I'm probably wrong. No. That's not correct. You do know what you're doing. You are smart enough to know that just because you have all this, I mean, like you were going toe to toe with Graham Short, asking him questions and saying things like that I didn't understand. Just to be clear, like you're using words that I don't know. Oh, the Coreys have this, that, and the other. You know what you're doing.
Yeah,
and
you know
and you know that seahorses need. So that's the key to me, is that I came from freshwater reefing than seahorses, and they're just different. None of them's harder than the other. In fact, I would say that raising and keeping discus and angelfish freshwater would be harder than seahorses any day of the week.
Yeah,
With
I
seahorses,
went easy freshwater to seahorses and didn't have a problem.
So I would say that with seahorses, it's just understanding that they need certain things. Good food, like Chris said, more feedings, like Ray always says, good water quality, like everybody says, and lower temps. So you can't keep everything you want to keep with them. You got to keep it under 74 degrees, or you should keep it under 74 degrees, unless you're talking about a huge tank, then it's different. Like there's all these caveats, right?
Yeah.
But here's the one thing you did right. The number one thing that a new person should do is come to Wine Wednesday. That's it. They need to come to Wine Wednesday. They need to come in Zen, wink, wink. They need to come in and actually, they don't have to show their face. They can do like Chris is doing right now, where he's here, he can talk, and he can show us maybe the tank, but not their face. And just, say, so this is what I've got going on so far. This is what I want to do. What do you guys think? We're not the end all. We're not, our answers aren't always 100% correct, but that's why we have this range of people. Ray, that's been doing it for 125 million years. Just kidding, Ray. And then Holly, who's bred, me, who's bred. We have different people from different areas that can come in and say, well, this is why I wouldn't suggest this, or why
I wonder if my DNA has been changing. over that
I don't know.
125 million years,
I know.
Probably, Ray.
I might be a separate species now.
Shut up,
You
Ray.
might. You might.
but anyways, I would say really seriously understanding that they're different, because I think
Mhm.
the most people that have the most trouble are people that have kept reefs or kept freshwater and think, oh, it's a smooth transition, it'll be the
Yeah.
same, they just need the same, especially reef keepers, right?
Yeah.
And I was guilty of that myself. That's why I'm saying it, is I ran into all sorts of trouble because I thought I could just, do things like a reef tank, and it's just a little bit different. The only other thing I would say for somebody setting up is pre-cycle. That's it. Have your tank cycled so you don't have to worry about ammonia or any of that crap. The nitrogen cycle, that's the
Mhm.
most important part of keeping any kind of tank. Have that taken care of ahead of time. Whether you want to use rock, K1, I don't care what you're using. Have that tank cycled to where it's cycled, so that you can focus on my next comment, observation. So different people have different thoughts about seahorses. My big thing is observation. If you observe them and you learn their habits, behaviors, and you learn who they are, I don't want to sound like
Yeah.
Holly, a seahorse mommy, but seriously, they have personalities.
Mhm.
That way you will know, okay, this seahorse isn't
Did you
sick.
see?
He's just pissed off at me, right? Or this seahorse isn't sick. She's just producing eggs or whatnot. It's learning that, and then having a resource. And I keep saying us, Wine Wednesday, but there's others. Having a resource, someone who's done it for a long time, to go to and say, because that was my key. The only reason I was successful is because I had Dan, I had Felicia McCulley, I had Cheryl, and I had Ray that I could go to and say, this seahorse all of a sudden has white spots, and they'd say, you're an idiot, that's coloration. And I'd be like, oh, okay. I mean,
Mhm.
or I would say my heater malfunctioned, and now this seahorse has a bunch of black spots and it looks like its skin's peeling off. And they'd say, okay, we need to get these medications and get in there. And that's what we do for you, and what any resource that you have does for you. Having that resource is really key. I know I'm rambling, guys. But there was one other thing I did want to say. Keep in mind setting up. I guess that's it. I know observation is not setting up, right? But for setting up, I would just say have the tank cycled completely before you even try for seahorses. And yeah, pre-think what you want to do and decide ahead of time and set up for it.
Mhm.
So, in other words, make sure you have the filtration methods to feed more, right? Or be prepared to do more water changes. There's always a caveat. You can do it
Different
this
ways to do
way.
it, but plan
Yeah,
ahead, I think, is key.
plan ahead.
Plan
That's
for
it. And you have. So stop saying you don't know what you're doing. You absolutely do.
them.
Something I'd like to
Please.
say to people when they're starting out, and especially if they're on forums or on Facebook pages and so on, go with what the majority of long term keepers recommend to
Mhm.
start. Once you've got your own experience, then you make decisions based on your experience, then
Yep.
what you want to branch out into or to try different. But if you're going to start off doing something that somebody says you can do and not many people do it, then you're asking for trouble.
Sure. No, I totally agree. Anytime I see an amazing seahorse tank, but I'm like, wait a minute, why do you have that fish in there?
Yeah.
Or why is that is that stinging coral in there or whatnot? I'll nicely just ask,
How long?
how long have you had those seahorses, right? Because anybody can set up a tank and take pictures and make it look beautiful. Some of the YouTubers, I won't mention names, but take a look. Some of the YouTubers that set up seahorse tanks, they just kind of never mentioned them again. That's key. So maybe don't listen to those. people. But yeah, if you see a post, maybe say, how long have you kept them? How's it going? Da da da. If it's not a year, yeah, I wouldn't follow in their footsteps. I would come here, send everybody to Wine Wednesday. That's it. Uh, and let's see. You thought you were prepared, but after this freak tornado, I've decided I will likely need a chiller that will be okay on a generator in the event of a power outage during summer again. Right. And listen, I sit here and say be prepared. I don't have a chiller. I don't have that. So the more prepared you can be, yes. But I also would say I wouldn't not set up because you're worried about a freak tornado, if that makes sense. Like
Yeah.
that's just looking for excuses not to do it.
I
And
mean,
I'm not
it
saying
took
that to
me a
you.
while to put together everything I had.
Yeah.
I
Yeah.
wouldn't worry
So
about the chiller for short-term needs. Like seahorses can handle
Yeah.
conditions like that. When you have long-term outages, so if you're out a couple days and the water's getting high, if you're able to salvage the system by keeping it aerated and so on, and get them through that, afterwards do a major water change, and they'll get through it.
Yep.
They are fairly resilient. They can
Mhm.
handle the warmer temperatures. It's if those warmer temperatures lead to an increase in bacterial growth
Yep.
that you don't rectify when as things become able to be rectified.
Mhm.
In other words, once your power comes back on, make sure then you're going to do a good complete water change, which all of a sudden knocks the hell out of the bacteria population. And bacteria, just for say you lost your power for 3 days or so, the bacterial levels are going to raise up, but it's not likely that they're going to get to do sufficient damage in those 3 days. So by doing that major water change afterwards, you're going to lower that population density of the bacteria right away and get back down to a more normal situation. And that gives the seahorses systems themselves to be able to fight off the remnants of the that slight increase that has happened.
So I just want to make a comment real quick because I'm so glad you said that, Ray, about the bacteria and just how to handle it and the big water change. Because I've talked to you guys many times. I have a video out there about dealing with the flesh eating bacteria that took over my seahorses. And what basically happened was I was, I think, out of town at work, something. My central air went out, and I don't have a chiller. I rely on my central air, and it went out, and the temp went from 70 to 72 to 82, like 10 degrees. Still wasn't a deal breaker, as Ray is saying. I could have salvaged the situation, but I didn't know what I was doing back then. And basically, I came home. I'm freaking out. It's all the temps way up. I'm like shit. So I lowered it really fast as I could with ice and such. One of the key things is if that happens to you, do it slowly. And the real key is do a huge water change. I did not do that. I did a normal water change. But if you think about it, what Ray just said, the temp spiked, bacteria grew crazy. I lowered it quickly, which messed with the seahorses. But basically, then the bacteria that was growing at that higher temp died. And now I've got all these organics just messing everything up. And the bacteria, dead or not, it's still causing problems. Had I done a major water change, I may have not have had problems. But
Mhm.
so anyways, drugstore peroxide can help the oxygen problem. Okay, wait, what are we talking about, Ray? Because I see your comments and I don't know what we're talking about.
Oxygen.
Yeah.
Okay,
Well,
we were out of, wait, hang on. We were out for 3 days this time, and 8 days was the longest. And this time I lost a bunch of hill stream loaches because I didn't have supplemental oxygen. So I just want to be extra prepared. I'm going to let you answer, Ray. But also, I would say with seahorses and their feeding, for you to take a vacation, you're going to have either live foods or somebody to come feed them. So you're going to have somebody who's going to notice within that 3 to 8 days. Go ahead, Ray.
Well, with the peroxide, you can use that peroxide to dump it into the tank,
Yep.
and that extra oxygen molecule helps to take care of any depletion of the oxygen that's happening. Now, mind you, you might have to use quite a bit if you don't have any other source, and it helps if you, even if you have to take a juice jug or something, and the larger the container, the better, and take out water and dump it back in. Take it out, dump it back in.
Yep.
You'll get a lot more oxygenation that way. But by adding that peroxide, that extra molecule can go a long ways to helping.
Sure. And I would say, first of all, I've done that before. The first time my power went out, I didn't even have seahorses. I had, I think it was discus and angelfish and all that. And I literally took a cup and I'm literally pouring it in, pouring it out, pouring it in. I wasn't concerned about temperature as much as oxygenation. And so that's definitely a key thing. And Holly, do you still hydrogen peroxide dose your tanks?
I do one of them, just one of them.
Okay. And
Yeah.
why?
Just trying to keep ciliates down.
So it keeps ciliates down. It creates that extra molecule, oxygenation. Peroxide is our friend in the seahorse world. So that's number one. That was the answer to your first question. Have a bottle of peroxide.
You do have to, if you're going to add a lot, you have to watch your salinity, though.
Sure. No, absolutely. I've never added a lot. I've only used it in the ways that we've talked about, but
I had to rescue one of Scott's goldfish once when we had a power outage because they weren't on anything. They weren't getting any oxygen or anything. And one of them was gasping and he's like, oh, they'll be all right. Because they come to the surface for air and blah, blah, blah. But then one of them started getting lopsided and we were losing her. And I told Scott, I'm like, you can't do that. So I took her and I put her in a dishpan of water, of clean water, and just ran it through her gills. I swished her around and then I put her in a bucket with an airline, from some of my stuff, and she was fine.
Holly's lying, you guys. She did mouth to mouth on the goldfish. She did. She pumped the chest. She did CPR. No, but no, I would like to put together like a list, and it always gets scary because I'm one of those hidden perfectionists where I like try to make it perfect and I never accomplish that goal. But I'd like to put together a simple list, like not everything you could possibly ever need to have to keep seahorses, but something simple like when you set up your seahorse tank, have prime on hand, have peroxide on hand, just the simple thing, not all the medicines and all that, but I'll work on it for sure. But Zen, you're capable. If you don't want to do it, nobody's pressuring you. But I know that you do want to do it, and we want you to do it, and you're going to succeed. So stop worrying. You've got not only the experience with just keeping fish, but you've listened to us forever. You've got us if there's a problem. I understand you're going through craziness with the plumbing and then the tornado and all that. No, there's no pressure. I know I sound like that. I don't mean to sound like that. I'm not pressuring you. But we want to see you succeed with them because the more people that do, the more people know. You know what I mean? You'll share too. So I hope that you believe in yourself because we, as Ray said, we all believe in you for sure. Hi, Chris. Anything else while we're live, or do we need to go to the after party?
We can do it either way. I was just going to say, like, Zen should definitely come on one week, and we can help guide her through it. We don't bite. We're all pretty friendly. Like, Graham was like, I was just talking to him behind the scenes. That's why I went off for a little bit, and I was just telling him, I was giving him some contact information to try and track down some read-eye for his research. But I was, he was just saying how nice everyone was, and how lovely it was. So hopefully we can even get him back somewhere down the line.
He's so great. I really like Graham. Like, I've always just really liked him as a person. But that's the other thing, is that he's so down to earth. He doesn't care if I don't understand what he's saying and ask a dumb question. He's gonna answer it and be polite and kind, and
Definitely.
it's great. I
Not all
love it.
Definitely. I mean, even I've asked him dumb questions, so don't stress.
Yeah, well, not as dumb as mine. But what, Ray? Ray?
Not all scientists are like him, believe me.
I know.
I run into so many, their heads are so big they can't get it.
They can't even talk.
It's hard to get in the house.
But that's why we're lucky that Graham, the Signathid person, is not like that. And he's hooking us up with Emily, and what was the other, Andy. And sorry guys, I'm terrible with names. But yeah, I think it'd be really awesome. So guys, I would say this though, because these people are very kind and just care, and they care about us in addition to sharing their research, right? So Graham comes even though we only had tops 10 people watching tonight, right? People will watch later, but it's not about that for them, right? They're really just trying to help and share. But if I get people like Emily and Andy, is it Andy? Am I saying the right name? Anyway, if I get these important people in here, we gotta share the posts so that we get more people, so that they feel like they made a difference by coming, right? They'll come anyways because they're just cool people and nice people, but it would be good for them to feel appreciated by having more viewers, if that makes sense. So if I get all these people together, you guys got to help me share around. Shyla, you started
speaking of sharing, did you see Shyla still has some of her babies?
No, where are we at with that? I'm sorry.
She has 9 left. They're about 4 months old. I think she said 3 to 4 months. It's gone off my screen. It was way earlier.
Okay, I'm sorry, guys. When I got a guest, I'm not very good at the comments. I apologize. Let's blame Holly. We'll blame Holly. Holly needs to be the comment person. No,
I'm going to have to leave anyway.
We were talking about Zen.
Wait. Hang on. Let's let Ray go. Hey, Ray, thank you for coming.
I've got to give my dog some time.
Go give the dog some time. We appreciate you coming and all your excellent questions and comments. Thank you, Ray. Have a good night.
Good night,
Good seeing you again, Chris.
Bye, Ray. Hey, likewise, mate.
And good news for all the people that have been missing Cheryl. She was trying to join all night.
Yeah.
But when I was clicking her, it wasn't letting her join. We had a technical problem, but she'll be back, which is really exciting. So anybody who missed Cheryl, she's going to hopefully be back next week. And yeah, I can't find the comment, but Shyla, that's really amazing that you're doing so well with them and that they are still doing well. I also see that you said that you guys don't have those natural disasters there. Lucky you. Every time it thunderstorms here, I'm like terrified there's going to be a tornado. I'm just so scared. I hate tornadoes. I don't know. Anyways, what else, guys? Anything else before we do end it?
I was just going to say that you've been doing really, really great with the channel lately. Like, you've had some real sort of real awesome, guest stars and guest appearances, and really killing it. It's good. Good job.
Thanks. So you guys, again, you guys just got to help me share on your pages, share in your groups, make sure people know to come so that we can grow it. But I would say too, again, my apologies for not having Aqua Balls do the tour. I didn't think he wanted to, or we didn't discuss it. I shouldn't say I didn't think he wanted to.
Yeah.
We didn't discuss it prior, so I didn't want to ask him while we were live.
Yeah.
And I'm last minute, like always. I'm always last minute. But now that I know that he will, I'm either going to have him on next week if Macro Lady can't come. Hopefully Macro Lady's going to come next week, but I haven't gotten a definite. If she doesn't, we'll have him back if he's available, show his tanks and all that stuff. So anytime there's something I miss or don't do, call me out so I can fix it. But all right, if that's it for tonight, guys, let's say Happy Wine Wednesday and start the after party. All right. I was hoping Cheryl would stick around for
I'm on the
that,
ice
but
too.
hey,
I
it
got
works.
water.
It works. I'm still on wine, guys, but I switched to red. All right.
Cheers.
Happy Wine Wednesday. See you next week, guys. I'll
Happy
post
Wine
whatever it's going to be.